Welcome to this exploration of neuroscience, designed for learning at your own pace. Whether you're actively listening or letting the information wash over you as you fall asleep. There's no test waiting at the end, no pressure to remember everything. This is simply an invitation to understand the most complex structure in the known universe, your brain. If you find this helpful, consider subscribing for more content on science, history, and the natural world. And we'd love to know, where are you listening from tonight? Drop your city or country in the comments below. It's always fascinating to see how curious minds connect from across the globe. Now, find a comfortable position. Let your body settle. Close your eyes if you'd like. And let's begin our journey through the brain, exploring how 86 billion neurons working together create everything you think, feel, remember, and experience. Let's discover how you work. The nervous system landscape. You are conscious right now. You're aware of these words, understanding their meaning, perhaps forming opinions about them, experiencing the sensation of thought itself. You know what it's like to be you. You have a perspective, a point of view, an inner experience. This should not be possible. Neurons are cells. Cells are made of molecules. Molecules follow the laws of chemistry, which reduce it to the laws of physics. Nowhere in physics is there an equation for consciousness. Nowhere in chemistry is there a formula for subjective experience. And yet somehow, purely from the mechanical interactions of neurons firing in patterns, awareness emerges. Thought, feeling, the sense of being. For centuries, this seemed to require something beyond physical law, a soul, a spirit, some special non-material ingredient that animated mere matter into conscious experience. Descartes, in the 17th century, was so convinced consciousness couldn't arise from physical processes that he proposed dualism. The mind and body are separate substances, interacting through the pineal gland deep in the brain. The physical body follows mechanical laws. The immaterial mind experiences and thinks. Somehow, impossibly, they communicate. This satisfied almost no one, even at the time. How could an immaterial mind push on material neurons? How could physical damage to the brain, which everyone knew altered mental function, affect an immaterial substance? Dualism solved nothing. It just renamed the mystery. By the 19th century, evidence was mounting that consciousness was physical. Stroke victims with damage to specific brain regions lost specific abilities. Speech, comprehension, memory, personality, all could be altered or destroyed by physical lesions. The mind wasn't hovering somewhere above the brain. It was the brain, or at least it arose from the brain's activity. But this created a new problem. If consciousness is physical, if it's just neurons doing what neurons do, following physical laws with no awareness or intention, how does experience happen? How do you get from electrochemical signals to the feeling of pain, the redness of red, the taste of coffee? How does subjective experience emerge from objective processes? This is called the hard problem of consciousness, and it remains unsolved. We've made enormous progress understanding the brain. We can map which regions activate during different tasks. We can watch neurons fire in real time. We can identify the neural correlates of consciousness, the specific patterns of activity that correspond to conscious states. But correlation isn't explanation. We still don't know how or why neural activity produces subjective experience. We're going to spend this lecture exploring what we do know about the brain and nervous system, how it's organized, how neurons work, how information flows through neural circuits. By the end, you'll understand the machinery. But fair warning, understanding the machinery doesn't solve the hard problem. We'll have explained how the brain processes information, stores memories, generates behavior. We won't have explained consciousness itself. That remains mysterious. Still, the machinery is remarkable enough to warrant attention. So let's begin with the landscape, the overall architecture of the nervous system, before we examine how the components work. The nervous system divides into two major parts, central and peripheral. The central nervous system, brain and spinal cord, is where processing happens. The peripheral nervous system, all the nerves branching throughout your body, carries information in and out. This division isn't just convenient labeling. It reflects fundamental differences in function and vulnerability. The central nervous system is protected. Your brain sits inside your skull. Your spinal cord runs through your vertebral column. Both are cushioned by cerebrospinal fluid and wrapped in protective membranes. There's also the blood-brain barrier, a selective filter preventing most substances in blood from entering brain tissue. This protects against toxins but makes delivering drugs to the brain notoriously difficult. Why so much protection? Because the central nervous system doesn't heal. Damage a peripheral nerve, and it might regrow. Slowly, imperfectly, but sometimes successfully. Damage your brain or spinal cord, and those neurons are gone, permanently. There's no backup, no regeneration. This is why strokes and spinal injuries are so devastating. The brain cannot repair itself the way skin or bone can. The peripheral nervous system has more flexibility. These nerves can sometimes regrow. They're also numerous, branching everywhere, carrying constant streams of information. Sensory nerves report on touch, temperature, pain, limb position, organ states. Motor nerves carry commands to muscles and glands. The peripheral system is the interface between your brain and the world. It divides into somatic and autonomic branches. The somatic system handles voluntary movement and conscious sensation. When you decide to move, somatic motor neurons execute the command. When you feel pressure or texture, somatic sensory neurons carry that information inward. The autonomic system operates automatically. You don't consciously control your heartbeat, digestion, or pupil dilation. The autonomic system manages these processes continuously without requiring attention, which is fortunate. Because if you had to consciously maintain your heart rate, you'd die the moment you got distracted. The autonomic system has two divisions. Sympathetic activation triggers stress responses. Increased heart rate, dilated airways, redirected blood flow, adrenaline release, fight or flight. Parasympathetic activation promotes rest and recovery. Decreased heart rate, active digestion, energy conservation. Rest and digest. Both are necessary. Problems arise when one chronically dominates, when stress keeps you perpetually activated, or when recovery mechanisms become sluggish. Now, let's explore the brain itself, because this is where things get interesting. At the base of the brain, where it connects to the spinal cord, you find the brainstem, evolutionarily ancient. Even reptiles have similar structures. The brainstem handles fundamentals: breathing, heart rate, blood pressure, arousal. These are the processes that keep you alive, operating continuously below conscious awareness. The brainstem has three regions. The medulla oblongata at the bottom manages vital autonomic functions. Damage here is often fatal. The pons, sitting above the medulla, relays information between brain regions and helps regulate sleep. The midbrain, at the top, processes sensory information and coordinates movement. Behind the brainstem sits the cerebellum, the little brain. It looks like a miniature brain tucked beneath the larger cerebral hemispheres, densely folded and packed with neurons. More neurons than the rest of the brain combined, actually, crammed into that relatively small space. For decades, neuroscientists thought the cerebellum just coordinated movement. People with cerebellar damage have tremors and poor motor control, so this seemed obvious. But the cerebellum does more. Recent research suggests it contributes to language, attention, emotion, possibly aspects of thought itself. Why pack so many neurons into a structure supposedly just for motor coordination? The answer is still emerging, but the cerebellum appears to be a general-purpose processor, fine-tuning not just movements but cognitive operations as well. Deep inside the brain lies the limbic system, structures involved in emotion, motivation, memory, and autonomic regulation. This is sometimes called the mammalian brain because these structures are well developed in mammals but less prominent in reptiles. The hippocampus, shaped like a seahorse buried in the temporal lobe, is essential for forming new long-term memories. Damage it, and you lose the ability to create new memories. You remember childhood, retain old skills, but new experiences don't stick. Yesterday never becomes memory. Every morning is unfamiliar. It's a devastating condition, first documented thoroughly in a patient known as HM, whose hippocampus was surgically removed to treat epilepsy in 1953. The surgery stopped the seizures, but destroyed his ability to form new memories. He lived the rest of his life in a perpetual present, unable to remember people he'd met minutes before. The amygdala, almond-shaped and near the hippocampus, processes emotions, particularly fear. Your amygdala constantly scans for threats, triggering fear responses before conscious awareness. You jump at shadows before recognizing them as harmless. The amygdala reacts first. Conscious recognition comes later. This makes evolutionary sense. Better to startle at nothing than fail to startle at danger. The hypothalamus, tiny but powerful, regulates hunger, thirst, body temperature, circadian rhythms, hormone release. It links the nervous and endocrine systems, translating neural signals into hormonal instructions affecting your entire body. The thalamus sits centrally, relaying sensory information. Almost everything you sense, except smell, passes through the thalamus en route to the cerebral cortex. It's a switchboard, routing incoming signals to appropriate processing centers. Finally, the cerebrum, the largest part of your brain, the wrinkled gray matter most people picture. The cerebrum divides into left and right hemispheres connected by the corpus callosum, a massive bundle of nerve fibers. The surface is elaborately folded into gyri and sulci, dramatically increasing surface area and packing more cortex into your skull's limited space. The cerebral cortex, the outermost layer, is where conscious experience happens. Thought, language, perception, voluntary movement, planning, reasoning. The cortex is what makes human cognition distinctive. The The cortex divides into 4 lobes per hemisphere. The frontal lobes behind your forehead handle motor control, speech, planning, decision-making, personality, emotional regulation. The prefrontal cortex, the most anterior part, manages executive functions, weighing consequences, inhibiting impulses, working toward goals. This region doesn't fully mature until the mid-20s, which might explain questionable adolescent decisions. The parietal lobes, behind the frontal lobes, process sensory information from your body: touch, temperature, pain, spatial awareness. They also handle navigation and spatial reasoning. Damage here produces strange symptoms. Some patients develop neglect, failing to notice one entire side of space, sometimes denying that half their body belongs to them. The condition seems impossible until you witness it. A person eating only food on the right side of their plate, insisting the left side doesn't exist, isn't being stubborn. Their brain genuinely cannot attend to that side of space. The temporal lobes, roughly behind your temples, process sound and store certain memories. Deep inside sit the hippocampus and amygdala. The left temporal lobe, in most people, is crucial for understanding language. Both temporal lobes help you recognize faces and objects. The occipital lobes, at the back of your head, handle vision. Light hits your retina. Signals travel to the occipital cortex and processing begins. Edges, colors, motion, shapes, faces, objects, all extracted through successive stages of analysis. Vision seems instant and effortless, but requires vast computational resources. Roughly a third of your cortex participates in visual processing somehow. Here's where things get strange. These regions process different aspects of vision separately. Color in one area, motion in another, shape in a third. This was discovered gradually through studies of brain-damaged patients who lost specific abilities while retaining others. Some patients lost color vision, but could still see shapes and motion. Others lost motion perception but retained color and form. The visual system is specialized, dissecting vision into components processed in different locations. But you don't experience vision as separate components. You see unified objects with color, shape, and motion integrated seamlessly. How? How does the brain bind these separate pieces into coherent experience? This is called the binding problem, and it's not fully solved. Neurons processing color don't directly connect to neurons processing motion. Yet somehow the information combines into unified perception. Synchrony might be part of the answer. Neurons firing in coordinated rhythms binding related information. But the details remain unclear. This reflects a broader mystery in neuroscience. The brain is massively distributed. No single region does anything alone. Vision doesn't happen only in occipital cortex. Memory doesn't reside only in the hippocampus. Language doesn't exist only in temporal and frontal regions. Every function involves networks spanning multiple areas. Information flows constantly between regions through white matter tracks, the bundles of myelinated axons connecting different brain areas. White matter is as important as the gray matter everyone focuses on. Damage the wiring, and regions can't communicate even if individually intact. This is what happens in multiple sclerosis, where myelin degrades, disrupting communication between brain regions and producing cognitive and motor impairments. The brain also contains fluid-filled ventricles producing cerebrospinal fluid. This liquid, cushions the brain, removes waste, delivers nutrients. It circulates through ventricles and around the brain and spinal cord, providing buoyancy that reduces effective brain weight from 3 pounds to ounces, preventing the brain from crushing itself. The spinal cord extends from the brainstem through your vertebral column. Gray matter in an H-shaped core, white matter surrounding it, carrying signals to and from the brain. 31 pairs of spinal nerves branch out, connecting to torso and limbs. This is the landscape, complex but organized. Regions specialized for different functions but all interconnected. Ancient structures handling survival, intermediate structures managing emotion and memory, cortex processing conscious experience. All working together, producing thought, perception, action. The continuous experience of being you. The machinery is physical, cells following electrochemical laws. But somehow, from this machinery, consciousness emerges. We understand the hardware. We're still puzzled by how hardware produces experience. That's the mystery we're carrying forward as we zoom in to examine how neurons actually work. The cellular foundation. Here's a question that plagued neuroscientists for decades. What is the brain made of? It sounds simple. The answer should be obvious. But for most of the 19th century, it wasn't. Scientists could see brain tissue under microscopes, but what they saw was a confusing tangle. Fibers everywhere, branching and interconnecting, forming what looked like a continuous mesh. Some scientists proposed that the nervous system was a reticulum, a network where everything connected to everything else with no clear boundaries between components. The brain was one giant continuous structure, like a sponge, with information flowing freely through an unbroken web. This was called reticular theory, and it was supported by some of the most prominent neuroscientists of the era, including Camillo Golgi, who had developed a revolutionary staining technique that made individual neural structures visible under the microscope for the first time. Golgi's method used silver chromate to stain tissue, and for reasons that still aren't fully understood, it randomly labeled only about 1% of neurons, allowing individual cells to stand out against the background. Without this technique, brain tissue looks like an impenetrable thicket. With it, you could see individual structures. And what Golgi saw convinced him the brain was continuous. The fibers seemed to merge and blend. There were no clear boundaries. The reticulum theory seemed confirmed. Santiago Ramón y Cajal, a Spanish neuroanatomist, used Golgi's own staining method to reach the opposite conclusion. Cajal examined the same tissue, applied the same technique, and saw something different. He saw individual cells, distinct neurons with cell bodies, branching extensions, and what looked like contact points rather than true continuity. The neurons came close to each other but didn't merge. They remained separate entities communicating across gaps. This was neuron doctrine. The nervous system is made of discrete cells, not a continuous web. Cajal spent years documenting this, drawing meticulous illustrations of neurons in different brain regions, showing their diversity and distinctive structures. His drawings are still used in textbooks today, works of art as well as science. The debate raged for decades. Golgi versus Cajal, reticulum versus neuron doctrine. In 1906, they shared the Nobel Prize in Physiology or Medicine, despite holding completely contradictory views about the basic organization of the nervous system. Golgi used his Nobel lecture to attack neuron doctrine and defend the reticulum theory. Kajal was right, but even winning the Nobel Prize alongside him didn't settle the argument. The definitive proof came with electron microscopy in the 1950s, when scientists could finally see the gaps between neurons. The tiny spaces where one neuron's extension came close to another without touching. These gaps were called synapses, and their existence proved neuron doctrine correct. The nervous system is cellular, discrete neurons communicating across synaptic gaps, not a continuous reticulum, but a network of individual components. This matters more than you might think. If the brain were a reticulum, information would flow freely everywhere with no clear pathways or boundaries. Specificity would be impossible. But if the brain is cellular, with neurons communicating through specific synaptic connections, then information can be routed through defined circuits. Specificity becomes possible. Learning can occur by strengthening or weakening specific synapses. The cellular nature of the nervous system is what makes it capable of the precision and flexibility we observe. So, what is a neuron? Fundamentally, it's a cell. It has a nucleus containing DNA, mitochondria producing energy, all the standard cellular machinery. But neurons are specialized for a specific function, transmitting information through electrical and chemical signals. This specialization has shaped their structure in distinctive ways. A typical neuron has three main parts. The soma, or cell body, contains the nucleus and most of the cell's metabolic machinery. This is where proteins are synthesized, where energy is produced, where the cell's genetic instructions are stored and executed. The soma ranges from about 4 to 100 micrometers in diameter, depending on neuron type. Extending from the soma are dendrites, branching structures that receive input from other neurons. The name comes from the Greek word for tree because dendrites branch like tree limbs, sometimes elaborately, creating what's called a dendritic tree. These branches are covered with tiny protrusions, called dendritic spines, specialized structures where synapses form. A single neuron might have thousands of spines, each receiving input from a different neuron. This is where information flows into the neuron. The axon is the neuron's output cable, a long, thin extension that carries signals away from the soma toward other cells. Axons can be short, just a few micrometers, or extraordinarily long. The neurons controlling your big toe have axons extending from your spinal cord down your leg, about a meter in length. In giraffes, some axons are several meters long. In blue whales, they can exceed 30 meters. The axon begins at a specialized region called the axon hillock, where the decision to fire or not fire is made. At its far end, the axon typically branches into multiple terminals, each forming a synapse with another cell. A single neuron might synapse onto thousands of other neurons, distributing its signal widely. Many axons are wrapped in myelin, a fatty insulation produced by specialized cells called oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. Myelin isn't continuous. It's laid down in segments with small gaps called nodes of Ranvier. This segmented insulation dramatically increases the speed at which signals travel down the axon, for reasons we'll explore when we discuss action potentials. This basic structure, soma, dendrites, axon, appears across billions of neurons, but there's enormous diversity in the details. Some neurons have elaborate dendritic trees. Others have sparse simple branches. Some have long axons. Others have short axons or no axon at all. Some are tiny. Others are comparatively giant. The variety is staggering, and neuroscientists have classified neurons into dozens of types based on structure, location, and function. Functionally, neurons fall into three broad categories. Sensory neurons carry information into the nervous system from sensory receptors. These are afferent neurons, a term meaning carrying toward. When you touch something hot, sensory neurons in your skin detect the temperature and send signals into your spinal cord and brain. Motor neurons carry information out from the nervous system to muscles and glands. These are efferent neurons, meaning carrying away. When you decide to move your hand, motor neurons carry that command from your brain and spinal cord to the muscles. Interneurons connect neurons within the central nervous system. They're by far the most numerous type, vastly outnumbering sensory and motor neurons. Interneurons process information, integrate signals, form circuits, and create the complexity that makes sophisticated brain function possible. When you think, remember, feel, interneurons are doing most of the work. Neurons aren't alone in the brain. They're surrounded by glial cells, which outnumber neurons roughly 1-to-1, though exact ratios vary by region. For a long time, neuroscientists dismissed glia as mere support cells, passive components that held neurons in place and provided nutrients. The name glia comes from the Greek word for glue, reflecting this unappreciated status. We now know glia do far more than provide structural support. There are several types, each with important functions. Astrocytes, star-shaped cells, maintain the chemical environment around neurons, regulating levels of ions and neurotransmitters. They're in close contact with both neurons and blood vessels, forming part of the blood-brain barrier. They also provide energy substrates to neurons and appear to participate in information processing in ways we're still discovering. Some research suggests astrocytes actively modulate synaptic transmission, potentially influencing learning and memory. Oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system produce myelin, wrapping axons in insulating layers. Without myelin, neural communication slows dramatically. Multiple sclerosis, where the immune system attacks myelin, demonstrates how critical this insulation is. As myelin degrades, signals become delayed or blocked, producing motor and cognitive impairments. Microglia are the brain's immune cells, constantly surveying for damage, pathogens, or debris. They're not derived from neural tissue but migrate into the brain during development from the immune system. When they detect problems, microglia become activated, changing shape and releasing signaling molecules. They can prune synapses during development and in response to learning, suggesting they play a role in circuit refinement. Ependymal cells line the ventricles and produce cerebrospinal fluid, contributing to waste removal and nutrient delivery. For decades, the neuron-centric view dominated neuroscience. Neurons were the stars. Glia were supporting cast. But it's increasingly clear that glia are active participants in brain function. The old metaphor of neurons as computers and glia as power supply no longer holds. Both are essential. Both contribute to information processing, The brain is not just a network of neurons, but a complex tissue where multiple cell types interact. Here's where the cellular foundation connects to a deeper puzzle. Neurons are cells. Cells are chemistry. Chemistry follows physical laws with no room for intention or awareness. And yet, from the coordinated activity of billions of these cells, consciousness emerges. Thought, experience, the sense of being. This isn't vitalism, the discredited idea that living things contain some special non-physical life force. Neurons follow the same laws of physics and chemistry as any other cells. There's no magic ingredient, no special consciousness particle. The mystery is how subjective experience arises from objective processes, how chemical reactions become thoughts. We can describe the machinery in increasing detail. We can map which neurons connect to which, trace the flow of signals through circuits, identify the chemical transmitters used at each synapse. But describing the machinery doesn't explain consciousness any more than describing a computer's circuitry explains why certain computations feel like anything from the inside. Some philosophers and neuroscientists argue this is a category error, that we're asking the wrong question. Consciousness, they suggest, is what information processing feels like from the inside. It's not something additional to neural activity, but identical with it, seen from a different perspective. The external, objective view sees neurons firing. The internal, subjective view is the experience those neurons produce. There's only one thing happening, described two ways. Others argue this merely restates the problem without solving it. Why should some information processing systems have subjective experience while others don't? Your brain apparently has consciousness. Your liver, which also processes information in its way, presumably doesn't. What's the difference? Complexity? Organization? Some specific type of information processing? The answer remains elusive. For now, we can bracket the hard problem and focus on the machinery itself, which is remarkable enough to warrant attention. Neurons communicate through electrical and chemical signals, forming circuits capable of extraordinary computational feats. They adapt and change in response to experience, enabling learning and memory. They maintain themselves over decades, preserving identity and knowledge even as individual molecules are replaced. The cellular foundation, neurons and glia working together, provides the substrate for everything the brain does. Perception, action, thought, emotion, memory, all emerge from the coordinated activity of these cells. Understanding how they work, how they communicate, how they form functional circuits, gets us closer to understanding how the brain produces mind. It doesn't fully solve the mystery. The hard problem of consciousness remains, but it reveals the machinery underlying mental life, and that machinery, once you understand it, is astonishing in its elegance and complexity. The neuron is not just a cell. It's a computational element, capable of integrating thousands of inputs, making decisions about whether to fire, transmitting signals rapidly over long distances, forming specific connections with precise targets. It's adaptable, changing its properties based on experience. It's efficient, operating on minimal energy. And there are billions of them, interconnected in ways we're still mapping. This is the cellular foundation. Now, let's see how these cells actually communicate. How electrical and chemical signals flow through neural circuits to produce the functions we associate with the nervous system, and ultimately with mind itself. Neural communication. In 1791, Luigi Galvani, an Italian physician and physicist, was dissecting a frog in his laboratory when he noticed something strange. When his metal scalpel touched the frog's nerve while the leg was in contact with a different metal surface, the dead frog's leg twitched. It jerked as if alive, muscles contracting violently. Galvani believed he had discovered animal electricity, some vital force unique to living tissue. He was partially right and fundamentally wrong. There was electricity involved, but there was nothing mystical about it. The frog's neurons and muscles, though dead, retained enough of their chemical gradients that the metal contact created a circuit, triggering the same electrical signals that would have occurred in life. Alessandro Volta, another Italian scientist, disagreed with Galvani's interpretation. Volta believed the electricity came from the contact between dissimilar metals, not from any special property of living tissue. He was also partially right. The metals did generate current. But Galvani was correct that nerves used electricity. Both men were seeing different aspects of the same phenomenon. The debate sparked decades of research into what became known as galvanism, the study of electrical effects in living tissue. Scientists of the era conducted increasingly bizarre experiments, applying electrical currents to dead animals to recently executed criminals, making corpses twitch and grimace in ways that horrified and fascinated public audiences. Mary Shelley drew on these experiments for Frankenstein, published in 1818, where a scientist reanimates dead tissue through electrical means. But despite all this experimentation, the mechanism remained mysterious. How did nerves actually use electricity? Were they like wires conducting current? That seemed unlikely because nerves conduct signals far too slowly. Electricity in copper wire travels near the speed of light. Signals in nerves travel at maybe 100 meters per second, less than the speed of sound. If nerves were simple conductors, signals should be nearly instantaneous. They're not. The solution wouldn't come for over a century, and when it did, it revealed something more elegant and complex than simple electrical conduction. Neural communication involves a sophisticated interplay of electricity and chemistry. Membranes and ions, voltage changes and molecular pumps, all coordinated in ways that make neurons capable of rapid, reliable, and flexible information transmission. Let's start with what a neuron is doing when it's not doing anything. Even at rest, a neuron maintains an electrical potential difference across its membrane. The inside of the cell is about negative 70 millivolts compared to the outside. This is called the resting potential, and maintaining it requires constant work. The membrane is a lipid bilayer, a thin barrier of fat molecules that separates the inside of the cell from the outside. This barrier is largely impermeable to charged particles, ions, which is crucial because the inside and outside of the neuron have very different ion concentrations. Outside the cell, sodium ions are abundant. Inside, they're scarce. Potassium ions show the opposite pattern. Abundant inside, scarce outside. This concentration difference is not random. It's maintained by sodium-potassium pumps embedded in the membrane. Molecular machines that use energy from ATP to continuously pump sodium out and potassium in. 3 sodium ions out for every 2 potassium ions in. This creates both a concentration gradient and an electrical gradient. sodium ions want to flow into the cell, driven by both their concentration difference and the electrical attraction to the negatively charged interior. Potassium ions want to flow out, driven by their concentration difference, though the electrical gradient pulls them inward. At rest, the membrane is slightly permeable to potassium, but mostly impermeable to sodium. Potassium can leak out through specialized channels, and it does, following its concentration gradient. But as it leaves, it carries positive charge with it, making the inside of the cell more negative. This negative charge starts to pull potassium back in. Eventually, an equilibrium is reached where the electrical pull inward balances the concentration-driven diffusion outward. That equilibrium is the resting potential, around -70 millivolts. This is the resting state. The neuron is loaded, like a spring compressed and ready to release. It's not passive. It's actively maintained in a high-energy state through constant work by the sodium-potassium pumps. About 20% of your brain's energy consumption goes to maintaining these gradients across billions of neurons. Now, what happens when a neuron fires? The signal begins at the dendrites and soma, where the neuron receives input from other neurons. Each input is small, a tiny change in voltage. Some inputs are excitatory, making the membrane potential slightly less negative, pushing it toward the threshold for firing. Others are inhibitory, making the membrane potential more negative, pushing it away from threshold. The neuron integrates these inputs, summing them spatially and temporally. Inputs arriving at the same time add together. Inputs close together on the dendritic tree influence each other more than distant inputs. The neuron is performing a calculation, weighing thousands of inputs to determine whether the combined effect exceeds threshold. If the membrane potential at the axon hillock reaches about -55 millivolts, something dramatic happens. Voltage-gated sodium channels open. These are protein pores in the membrane that sense voltage and change shape in response. When the membrane depolarizes to threshold, these channels snap open, and sodium floods into the cell, driven by both its concentration gradient and the electrical gradient. This influx of positive sodium ions further depolarizes the membrane, which opens more sodium channels, which brings in more sodium, which opens even more channels. It's a positive feedback loop, explosive and self-reinforcing. Within about 1 millisecond, the membrane potential shoots from -55 millivolts to +40 millivolts. This rapid reversal of membrane potential is called an action potential. But it doesn't last. Almost as soon as the sodium channels open, they begin to inactivate, a separate process from closing that renders them temporarily unresponsive. Simultaneously, voltage-gated potassium channels open, slower to respond than sodium channels but sensitive to the depolarization. Potassium rushes out of the cell, driven by both concentration gradient and now the electrical gradient, since the inside has become positive. This outflow of positive charge repolarizes the membrane, bringing it back toward rest. In fact, it overshoots slightly, becoming even more negative than the resting potential for a brief moment. This is called hyperpolarization, and it serves an important function. During this refractory period, the neuron cannot fire another action potential. The sodium channels are inactivated and won't reopen until the membrane returns to resting potential. This ensures that action potentials travel in one direction, away from the soma toward the axon terminals, and it sets a limit on how fast a neuron can fire. Typically, no more than about 1,000 times per second. The action potential, once initiated, propagates down the axon. The depolarization at one location opens sodium channels in the adjacent membrane, triggering another action potential there, which triggers the next section, and so on. The signal moves like a wave, self-regenerating as it goes. In unmyelinated axons, this is relatively slow, about 1 meter per second. In myelinated axons, the signal jumps between nodes of Ranvier, the gaps in the myelin sheath. The myelin insulates the membrane, preventing ion flow. Only at the nodes can ions cross, so the action potential jumps from node to node, a process called saltatory conduction, from the Latin word for jump. This is far faster, up to 100 meters per second or more. This is how Galvani's frog leg twitched. The metal contact created a current that depolarized neurons past threshold, triggering action potentials that propagated to muscles, causing contraction. But the signal wasn't electricity flowing through a wire. It was a wave of ion channel openings, a chemical electrical cascade that propagated down the axon. This was worked out primarily in the 1940s and 1950s by Alan Hodgkin and Andrew Huxley, working with the giant axon of the squid, which is large enough to insert electrodes and measure voltage changes directly. They developed mathematical equations describing the action potential in such detail that they could predict its exact shape and propagation speed. This work won them the Nobel Prize in 1963 and remains one of the triumphs of quantitative neuroscience. But action potentials are only half the story. They carry signals within a neuron, from dendrites and soma down the axon. To communicate between neurons, to cross the synaptic gap, requires a different mechanism, chemical transmission. When an action potential reaches the axon terminal, it opens voltage-gated calcium channels. Calcium floods in, and this calcium influx triggers a cascade. Synaptic vesicles, small spheres containing neurotransmitter molecules, fuse with the membrane and release their contents into the synaptic cleft, the tiny gap between the presynaptic terminal and the postsynaptic neuron. Neurotransmitters diffuse across this gap, typically 20 to 40 nanometers wide, and bind to receptors on the postsynaptic membrane. These receptors are often ligand-gated ion channels, protein pores that open when the right molecule binds. Depending on the neurotransmitter and receptor, the result is either excitatory or inhibitory. Glutamate, the most common excitatory neurotransmitter in the brain, opens channels that let sodium in, depolarizing the postsynaptic neuron and increasing the likelihood it will fire. GABA, the most common inhibitory neurotransmitter, opens channels that let chloride in or potassium out, hyperpolarizing the postsynaptic neuron and decreasing the likelihood of firing. There are dozens of neurotransmitters, each with specific roles. Acetylcholine, used at the neuromuscular junction, also functions in the brain for attention and arousal. Dopamine modulates reward, motivation, and motor control. Serotonin regulates mood, appetite, and sleep. Norepinephrine affects alertness and arousal. Each acts on multiple receptor types, creating diverse effects depending on location and context. After neurotransmitters bind and exert their effect, they must be cleared from the synaptic cleft. Some are broken down by enzymes. Acetylcholine is cleaved by acetylcholinesterase, for instance. Others are taken back up into the presynaptic terminal through transporter proteins, recycled for future use. This clearance is crucial. If neurotransmitters lingered, signaling would become blurred and uncontrolled. Many drugs target this system. SSRIs, selective serotonin reuptake inhibitors used to treat depression, block serotonin transporters, leaving more serotonin in the synaptic cleft. Cocaine blocks dopamine reuptake, producing euphoria and reinforcement that can lead to addiction. Nerve gases block acetylcholinesterase, causing acetylcholine to accumulate, leading to overstimulation of muscles and glands, paralysis, and death. Chemical synapses are the dominant form of communication in the human brain, but there are also electrical synapses, gap junctions where the membranes of two neurons come into direct contact through specialized protein channels. Current can flow directly from one cell to another, no neurotransmitter required. These are faster than chemical synapses but less flexible. They're used where speed and synchrony are critical, such as in certain reflex circuits. Here's what makes this system remarkable. A single action potential is all or none. It either happens fully or not at all. There's no such thing as a small action potential or a large one. They're stereotyped, identical. This is called the all or none law, and it ensures reliable signal transmission. Action potentials don't degrade as they travel down axons. They regenerate at each point, maintaining full strength. But how does a system using all or none signals encode information? How does the brain represent the difference between a dim light and a bright one, a whisper and a shout? The answer is firing rate. A stronger stimulus triggers more action potentials per unit time. The information is in the frequency of firing, not the size of individual spikes. A neuron might fire a few times per second for a weak stimulus, hundreds of times per second for a strong one. There's also temporal coding. The precise timing of spikes can carry information. Neurons that fire together synchronously can have greater impact on their targets than neurons firing at random times. This is the basis of Hebbian learning, the principle that neurons that fire together wire together. Synapses between neurons that frequently fire in coordination strengthen over time, a mechanism underlying learning and memory. The synapse is also where plasticity occurs, where the nervous system adapts to experience. Synaptic strength can increase or decrease based on activity. This is synaptic plasticity, and it's fundamental to how the brain learns, how memories form, how skills develop. We'll explore this more when we discuss learning and memory, but for now, understand that the synapse is not fixed. It's dynamic, constantly adjusting its strength based on the patterns of signals flowing through it. This system, action potentials and synaptic transmission, is how information flows through the brain. Sensory input triggers action potentials in sensory neurons. Those signals propagate to the spinal cord and brain. Interneurons process the information, integrating inputs, forming patterns, making decisions. Motor neurons carry commands out to muscles, all of it mediated by the same basic mechanisms: voltage-gated channels, ion gradients, neurotransmitter release, receptor binding, its chemistry and physics, ions moving through protein channels, molecules diffusing across gaps, membranes charging and discharging. Nothing mystical. nothing that violates physical law. And yet, from this machinery, from billions of neurons communicating through trillions of synapses, thought emerges. Memory, perception, consciousness. We've described the mechanism. We understand the hardware. But how hardware produces experience remains the deepest mystery in neuroscience, perhaps in all of science. The action potential is elegant. Synaptic transmission is well characterized. But why it feels like anything from the inside, why there's subjective experience accompanying these objective processes that we still don't know. For now, we have the machinery. And it's time to see what this machinery can do, how it processes sensory information, controls movement, forms memories, generates emotion. The mechanisms we've described, action potentials and synapses, are the foundation. Everything else is built from these components, combined in circuits and networks that perform the astonishing computations underlying every aspect of mind. Sensory systems. Right now, as you read these words, you're experiencing what seems like direct contact with reality. You see the letters, perhaps hear sounds around you, feel the surface beneath you, maybe taste something lingering from your last meal. The world feels immediate, present, obvious. It's not. You're experiencing a simulation. Your brain has never directly encountered the external world. It sits in darkness inside your skull, sealed away from direct contact with light, sound, chemicals, or touch. Everything you experience is constructed from electrical signals, patterns of action potentials arriving from sensory receptors scattered throughout your body. The brain receives these signals, interprets them, and generates a model of reality. What you experience as the world is actually your brain's best guess about what's out there. This isn't philosophical speculation, it's neurological fact, and it creates a profound puzzle. How does the brain transform meaningless patterns of neural activity into the rich, coherent, meaningful experience of perception? Consider vision. Light reflects off objects, enters your eyes, strikes the retina at the back of each eyeball. The retina contains photoreceptor cells, rods, and cones, which convert light energy into electrical signals through a chemical process called phototransduction. Already, information has been translated from electromagnetic radiation into the language of neurons, action potentials, and graded potentials. But the retina doesn't just passively capture images like a camera. It pre-processes visual information, extracting features through several layers of cells. Bipolar cells receive input from photoreceptors. Horizontal cells and amacrine cells perform lateral processing, comparing signals from adjacent regions. Ganglion cells, whose axons form the optic nerve, integrate these signals and send them to the brain. Each ganglion cell responds to a specific region of visual space, its receptive field. Some cells respond best to light in the center of their field with darkness around it. Others prefer the opposite, darkness in the center with light surrounding. Still others detect movement in specific directions. The retina is already analyzing the visual scene, decomposing it into features before the information ever reaches the brain. The optic nerves from each eye meet at the optic chiasm, where axons from the nasal half of each retina cross to the opposite side of the brain, while axons from the temporal half remain on the same side. This crossing ensures that each side of the brain receives information from the opposite side of visual space. Everything to your left is processed by your right hemisphere. Everything to your right by your left hemisphere. Most visual information travels to the lateral geniculate nucleus of the thalamus, that central relay station we mentioned earlier. From there, signals project to the primary visual cortex, V1, located in the occipital lobe at the back of your brain. This is where conscious visual processing begins. V1 is organized retinotopically, meaning adjacent regions of the retina map to adjacent regions of cortex. There's a distorted but systematic correspondence between visual space and cortical space. The central region of your visual field, the fovea where detail is sharpest, occupies a disproportionately large area of V1. Peripheral vision is compressed into smaller cortical territories. This magnification of central vision reflects where our visual attention naturally focuses. Within V1, neurons are organized into columns, groups of cells with similar response properties. Some columns respond to vertical edges, others to horizontal edges or specific oblique angles. This was discovered by David Hubel and Torsten Wiesel in the 1960s through elegant experiments recording from individual neurons in cat visual cortex while presenting different visual stimuli. They found that neurons in V1 are edge detectors, responding to boundaries between light and dark at specific orientations. This discovery revolutionized neuroscience and earned Hubel and Wiesel the Nobel Prize in 1981. It revealed that the brain doesn't represent images as two-dimensional arrays of pixels. Instead, it extracts features, breaking down the visual scene into components that are then analyzed separately. From V1, visual information flows forward through a series of progressively more specialized areas. V2, V3, V4, V5, each extracting different features. V4 is particularly involved in color processing. Damage here can cause achromatopsia, the inability to perceive color. The world becomes grayscale, not because the eyes are damaged, but because the cortical area interpreting color signals is destroyed. V5, also called MT for middle temporal area, processes motion. Neurons here respond to movement in specific directions. Damage to V5 produces achinotopsia, motion blindness. Patients with this condition see the world as a series of static snapshots. Pouring tea becomes impossible because the stream of liquid appears frozen until the cup suddenly overflows. Walking across a street is terrifying because cars seem to teleport from place to place rather than moving smoothly. These cases reveal something crucial. Vision is not unitary. What you experience as seamless, unified visual perception is actually constructed from separate processing streams analyzing different aspects of the scene. Color, motion, form, depth, all are processed in different cortical areas. Yet you experience a single, coherent visual world. This is the binding problem we mentioned earlier. How does the brain integrate these separate features into unified objects? A red ball moving left should be perceived as one thing, not as disconnected attributes of redness, roundness, and leftward motion. The mechanisms underlying binding remain debated, though synchronized neural activity across regions appears to play a role. Vision also splits into two major processing streams after V1. The ventral stream. Flowing forward into the temporal lobe, processes object identity. What is it? This stream enables you to recognize faces, read words, identify objects. Damage here produces agnosias, failures of recognition, prosopagnosia, inability to recognize faces, results from damage to the fusiform face area in the ventral stream. Patients can see faces clearly but can't identify them, even their own reflection. The dorsal stream, flowing upward into the parietal lobe, processes spatial location and motion. Where is it? How is it moving? This stream guides action, enabling you to reach for objects, navigate space, track moving targets. Damage here can produce optic ataxia, inability to reach accurately for visible objects despite intact vision. These two streams, what and where, reveal that the brain doesn't construct a single representation of the visual world, but multiple representations optimized for different purposes. One for recognizing objects, another for guiding actions. Sometimes these representations can dissociate dramatically. Patients with a condition called blind sight have damage to V1, rendering them consciously blind in part of their visual field. Yet, if you throw a ball toward them, they can catch it. If you ask them to reach for an object in their blind field, they reach accurately. They claim not to see anything, and they genuinely don't have conscious visual experience. But information is getting through, processed by alternative pathways, sufficient to guide action even without conscious perception. This reveals something profound about consciousness. Visual information can be processed, can influence behavior without producing conscious experience. The action potentials are there. The processing is happening. But something is missing. Whatever ingredient transforms neural activity into subjective experience. Blind sight patients demonstrate that you can have vision without seeing, perception without experience. Hearing works through similar principles: transduction, preprocessing, hierarchical processing, feature extraction. Sound waves, pressure variations in air, enter the ear and vibrate the eardrum. These vibrations transfer to three tiny bones: the ossicles, which amplify the signal and transmit it to the cochlea, a fluid-filled spiral structure in the inner ear. Inside the cochlea, the basilar membrane vibrates in response to sound. Different frequencies cause maximal vibration at different locations along the membrane, creating a tonotopic map. High frequencies stimulate the base near the entrance. Low frequencies stimulate the apex at the far end. This is mechanical frequency analysis performed by the physical properties of the membrane before any neural processing occurs. Hair cells sitting on the basilar membrane detect these vibrations. When the membrane moves, tiny hair-like projections called stereocilia on top of these cells bend. This mechanical deflection opens ion channels, generating electrical signals that trigger neurotransmitter release onto auditory nerve fibers. Auditory information travels to the brainstem, where it's processed for sound localization. Your ability to determine where a sound comes from relies on comparing the signal arriving at each ear. Sounds from the left reach your left ear slightly before your right ear, and they're slightly louder in the left ear because your head blocks and absorbs some sound traveling to the far ear. These tiny differences, microseconds of timing difference and minimal intensity differences, are detected by specialized neurons in the brainstem that act as coincidence detectors, firing maximally when inputs from both ears arrive simultaneously. From the brainstem, auditory signals ascend to the medial geniculate nucleus of the thalamus, then to primary auditory cortex in the temporal lobe. Like visual cortex, auditory cortex is organized systematically. Adjacent regions respond to adjacent frequencies, creating a tonotopic map. Low frequencies are processed in one area, high frequencies in another, with smooth gradation in between. Beyond primary auditory cortex, processing streams diverge. One stream specializes in identifying sounds. What am I hearing? This includes speech comprehension, which in most people relies heavily on the left temporal lobe, particularly Wernicke's area. Damage here produces receptive aphasia, inability to understand spoken language. Patients speak fluently but nonsensically, producing grammatically correct sentences that convey no meaning, and they cannot comprehend what others say. Another stream processes sound location and movement. Where is the sound? How is it moving? This supports navigation, sound localization, and the ability to separate sounds from different sources, the cocktail party effect that lets you focus on one voice amid many. Touch, pain, and temperature are processed by the somatosensory system. Receptors in skin, muscles, joints, and internal organs detect mechanical deformation, temperature changes, and tissue damage. Different receptor types specialize in different stimuli. Mechanoreceptors respond to touch and pressure. Thermoreceptors detect temperature. Nociceptors signal pain. These sensory neurons send signals through the spinal cord to the thalamus, then to the primary somatosensory cortex in the parietal lobe, just behind the central sulcus that divides frontal and parietal lobes. This cortex is organized somatotopically, with a systematic map of the body surface. Adjacent regions of skin map to adjacent regions of cortex. But the map is distorted, reflecting not the physical size of body parts but their sensory importance. Your hands, particularly fingertips, occupy enormous cortical territory. Your lips and tongue are similarly magnified. Your back and legs, though physically large, occupy relatively little cortex. This distortion produces the famous sensory homunculus, a grotesque figure drawn to scale according to cortical representation rather than actual anatomy. The homunculus has massive hands, huge lips, and a tiny torso, reflecting the fact that some body regions have far greater sensory resolution than others. Taste and smell, the chemical senses, work through direct detection of molecules. Taste receptors on the tongue respond to 5 basic tastes: sweet, salty, sour, bitter, and umami. A savory taste detected by receptors sensitive to glutamate. Different regions of the tongue have slight preferences for different tastes, though the old myth of a strict taste map is false. All regions can detect all tastes. Olfactory receptors in the nasal epithelium detect airborne molecules. Humans have about 400 different olfactory receptor types, each responsive to different molecular features. The combination of receptors activated by a given molecule creates a pattern, a signature that the brain interprets as a specific smell. What's remarkable about olfaction is its direct connection to emotion and memory. Olfactory information bypasses the thalamus and projects directly to the olfactory cortex, which is part of the limbic system. This is why smells can trigger powerful emotional responses and vivid memories. The scent of a particular perfume might instantly transport you to a specific moment in your past with an intensity that visual or auditory cues rarely match. All of these sensory systems follow similar principles. Specialized receptors transduce physical or chemical stimuli into neural signals. Preprocessing extracts features. Information ascends through the thalamus to primary cortical areas. Higher cortical areas perform increasingly complex analysis. Parallel processing streams handle different aspects of the stimulus. And somehow, from this distributed processing, unified conscious experience emerges. You don't experience edge detectors firing in V1, color processing in V4, motion analysis in V5. You experience a red ball moving. You don't experience separate activation of auditory frequency analyzers. You hear a melody. The brain constructs perception from the raw materials of neural activity, transforming action potentials into experience. This is what your brain is doing right now. Photons hit your retina. Chemical reactions generate electrical signals. Those signals propagate through layers of processing, analyzed and interpreted, until they produce the experience of reading these words. The page or screen in front of you, the ambient light, the sounds around you, all are constructions, simulations generated by your brain from patterns of neural activity. The simulation is convincing. It feels real, immediate, direct. But it's mediated by millions of neurons performing computations you're not conscious of, applying assumptions and shortcuts you're not aware of, constructing a model of reality that's useful for survival even if it's not perfectly accurate. Perception is not passive reception. It's active construction. And that raises questions we still can't fully answer. How does construction become experience? How do objective neural processes produce subjective sensations? Why does red look like red? Why does middle C sound like middle C? These are aspects of the hard problem again, the mystery of consciousness lurking at the center of neuroscience. We've mapped the pathways. We've identified the neurons. We understand the mechanisms. But understanding how the brain processes sensory information doesn't explain why processing feels like anything. That gap between physical process and subjective experience remains. For now, we have the machinery, and the machinery, at least, is comprehensible, elegant, and extraordinary. Motor control and movement. In the 1860s, a German physician named Gustav Fritsch was treating wounded soldiers from the Austro-Prussian War. While cleaning a head wound, he accidentally touched exposed brain tissue with a metal probe. The soldier's body jerked. A muscle on the opposite side of the body had contracted, triggered by the contact with the brain. Fritsch realized he'd stumbled onto something significant. He teamed up with Eduard Hitzig, and together they began systematically stimulating the brains of dogs with electrical current, mapping which regions produced which movements. What they discovered was a motor map, a systematic representation of the body laid out across the surface of the frontal lobe. This was revolutionary. Before this, many scientists believed the cerebral cortex was functionally uniform, a general-purpose processor with no regional specialization. Fritsch and Hitzig showed that specific movements were controlled by specific cortical locations. Stimulate one spot, and the dog's leg twitched. Stimulate an adjacent spot, and the paw moved. The brain wasn't a homogeneous blob. It was organized, with distinct regions performing distinct functions. Later work by Wilder Penfield in the 1930s and 1940s refined this mapping in humans. Penfield was a neurosurgeon treating epilepsy by removing damaged brain tissue. Before surgery, he needed to map each patient's brain to avoid removing areas critical for movement or speech. With the patient awake under local anesthesia, he stimulated the cortex with electrodes, while the patient reported what they experienced. What Penfield found was a somatotopic map in the motor cortex, similar to the sensory homunculus we mentioned earlier. Different body parts were controlled by different cortical regions, arranged in a distorted representation running along the precentral gyrus just in front of the central sulcus. Feet at the top, near the midline. Then legs, trunk, arms, hands, face, lips, tongue, descending down the side of the brain. The distortion mirrored the sensory map. Body parts requiring fine motor control, hands and face particularly, occupied disproportionately large cortical territories. Your thumb alone has more motor cortex dedicated to it than your entire torso. This reflects functional importance, not physical size. You need precise control over your fingers and lips. Gross control over your back and legs is sufficient for most tasks. But motor cortex is just one component of a complex motor system. Movement isn't simply cortex sending commands to muscles. It involves multiple brain regions working in coordination, planning actions, selecting appropriate responses, refining execution, learning from errors. The motor system is hierarchically organized. At the highest level, the prefrontal cortex and posterior parietal cortex are involved in planning. What do I want to accomplish? What action would achieve that goal? These areas represent abstract intentions and goals, not specific movements. The premotor cortex and supplementary motor area, anterior to primary motor cortex, select and sequence specific movements. How do I execute this action? What sequence of muscle activations will achieve my goal? These areas are active before movement begins, preparing the motor system for action. Primary motor cortex then generates the actual motor commands. Neurons here project down through the corticospinal tract. A massive bundle of about 1 million axons descending from cortex through the brainstem and into the spinal cord. Most of these axons cross into the opposite side at the medulla, which is why the left hemisphere controls the right side of the body and vice versa. In the spinal cord, these descending commands synapse onto motor neurons that directly innervate muscles. Each motor neuron controls a group of muscle fibers, together called a motor unit. Small motor units, with just a few muscle fibers per neuron, allow fine control. Large motor units, with hundreds of fibers per neuron, generate strong but imprecise force. But this hierarchical, top-down view is incomplete. Two other brain structures play critical roles in motor control: the basal ganglia and the cerebellum. The basal ganglia are a collection of nuclei deep in the brain, including the caudate, putamen, globus pallidus, substantia nigra, and subthalamic nucleus. These structures don't directly command movement. Instead, they modulate activity in motor cortex, facilitating desired movements and suppressing unwanted ones. The circuit is complex, involving multiple loops and both excitatory and inhibitory pathways, but the functional result is action selection. The basal ganglia help choose which of many possible actions to execute and inhibit competing alternatives. When you reach for a cup, countless other movements are possible. The basal ganglia suppress those alternatives, allowing the selected action to proceed cleanly. Damage to the basal ganglia produces characteristic motor disorders. Parkinson's disease results from degeneration of dopamine-producing neurons in the substantia nigra. patients develop tremor, rigidity, and bradykinesia, slowness of movement. Initiating actions becomes difficult. Movements that should be automatic require conscious effort. The problem isn't paralysis. Muscles work fine. The issue is selecting and initiating movements, functions the basal ganglia normally facilitate. Huntington's disease, caused by degeneration of the striatum, produces the opposite problem. Excessive, uncontrolled movements called chorea. Random, jerky motions of limbs and face occur involuntarily. The basal ganglia's inhibitory control is lost, allowing unwanted movements to escape suppression. The cerebellum, that densely folded structure tucked behind the brainstem, receives copies of motor commands from motor cortex and sensory feedback about the results. It compares intended movement with actual movement, detecting errors and making corrections. The cerebellum is essentially a predictive error correction device, continuously adjusting motor output to match intentions. When you reach for an object, the cerebellum predicts where your hand will be based on the motor command sent. As your hand moves, sensory feedback reports its actual position. If there's a mismatch, if your hand isn't where the cerebellum predicted, corrective signals adjust the movement. This happens rapidly, within tens of milliseconds, far faster than conscious awareness. The cerebellum is also crucial for motor learning. When you practice a new skill, repeatedly attempting the same movement, the cerebellum gradually refines the motor program, reducing errors over time. Eventually, the movement becomes smooth and automatic. This is cerebellar learning, a form of supervised learning where errors drive adjustments. Damage to the cerebellum doesn't cause paralysis. Patients can move, but movements become clumsy, inaccurate, uncoordinated. Reaching for an object produces tremor that worsens as the hand approaches the target. Intention tremor because error correction is impaired. Walking becomes staggered and unsteady. Speech becomes slurred because the fine motor control required for articulation is disrupted. Motor control also involves reflexes, rapid automatic responses that bypass higher brain centers entirely. The simplest reflex arc involves just two neurons, a sensory neuron and a motor neuron, connected by a single synapse in the spinal cord. Touch a hot surface, and pain receptors send signals into the spinal cord. Those signals synapse directly onto motor neurons controlling arm muscles, triggering withdrawal. You pull your hand back before you're consciously aware of pain. The reflex protects you faster than conscious processing could. Most reflexes involve additional interneurons, creating more complex circuits, but the principle is the same. Rapid, automatic responses that don't require cortical involvement. This offloads routine protective responses from conscious control, freeing the cortex for more complex decisions. But here's what's strange about motor control. The process is far more complicated than it seems. When you decide to pick up a cup, that decision is conscious. You're aware of intending to move. But the actual execution, the precise coordination of dozens of muscles, the timing of their activation, the force each must generate, all of this is entirely unconscious. You don't know how you move your arm. You can't describe which muscles contract in which sequence. You just decide to move, and somehow your nervous system translates that abstract intention into coordinated motor output. The gap between intention and execution is filled with computations you are completely unaware of. Moreover, experiments have shown something deeply unsettling about voluntary movement. In the 1980s, Benjamin Libet conducted studies where subjects were asked to flex their wrist whenever they felt like it while he recorded brain activity. He asked them to note when they first became aware of the intention to move. What Libet found was that brain activity associated with movement, a slow electrical shift called the readiness potential, began about 550 milliseconds before the movement occurred. But subjects didn't report conscious awareness of intending to move until about 200 milliseconds before the movement. The brain was preparing to move before the person was consciously aware of deciding to move. This created enormous controversy. Does it mean free will is an illusion? That consciousness is just observing decisions the brain has already made unconsciously? The interpretation remains debated, but the basic finding is solid. Neural activity precedes conscious awareness of intention. One resolution is that consciousness isn't where decisions originate, but where they become reportable, where the brain's computations become accessible to verbal description and deliberation. The readiness potential might reflect unconscious preparation, with conscious awareness arising as the decision crosses some threshold of certainty or commitment. But this doesn't eliminate the puzzle. It just relocates it. What is consciousness doing if the brain can prepare actions before conscious awareness? Why do we experience intentions at all if the machinery works unconsciously? Motor control also reveals the brain's stunning adaptability. When people use tools, the brain incorporates them into body representation. Tennis players' rackets become, neurologically, extensions of their arms. The motor system treats the racket as part of the body, adjusting movements to account for its length and weight automatically. Even more dramatically, people can learn to control artificial limbs or computer cursors with brain activity alone. Brain-machine interfaces record neural activity in motor cortex and decode it to extract intended movements. Paralyzed patients have learned to control robotic arms, play video games, type messages, all by thinking about movement without actually moving. The motor cortex generates patterns of activity corresponding to intended movements. Algorithms decode these patterns and translate them into commands for external devices. After practice, users report that controlling the device feels natural, almost like controlling their own limbs. The brain adapts, treating the device as part of the body. This plasticity reveals something fundamental. The motor system isn't rigidly wired to specific muscles. It's flexible, learning arbitrary mappings between neural activity and outcomes. What matters isn't the specific muscles involved, but achieving intended goals. The brain can learn new embodiments, incorporate tools and prosthetics, adapt to injuries that alter which muscles are available. Consider phantom limb syndrome. Amputees often feel their missing limbs still present, sometimes painfully so. They can vividly sense the position of fingers that no longer exist, feel itches they cannot scratch. This occurs because the motor and sensory cortex representations of the limb remain intact even after the limb is lost. The neural machinery for controlling and sensing the limb persists, generating experiences of movement and sensation from a body part that no longer exists. Over time, the cortical representation can reorganize. The area formerly dedicated to the missing hand might become responsive to the face, which is represented adjacently in the somatotopic map. Some amputees report that touching their face produces sensations in their phantom hand, a result of this remapping. Movement also demonstrates the profound integration between brain and body. You are not a brain piloting a meat robot. You are an embodied nervous system. Your sense of self, your experience of agency, your perception of the world, all are shaped by your body's capabilities and the feedback it provides. Dance, sports, music, skilled crafts, all involve motor expertise that transcends conscious understanding. Athletes speak of muscle memory, though the memory isn't in muscles but in the motor circuits of the brain and spinal cord. Through practice, movements become automated, executed fluidly without conscious deliberation. This is procedural memory, knowledge of how to do things, distinct from declarative memory, knowledge of facts. You can ride a bicycle without being able to explain how you maintain balance. The knowledge is encoded in motor circuits, inaccessible to conscious reporting. Motor control is where mind meets world, where intentions become actions, where the brain's computations produce observable effects in the physical environment. It's the output side of the nervous system, the means by which internal neural processes influence external reality. And, like sensory processing, motor control is performed by mechanisms we can describe in detail. Neurons in motor cortex firing in patterns, signals descending through the spinal cord, motor neurons activating muscles, feedback loops refining execution. All of it physical, chemical, electrical. Yet the experience of willing a movement, of being the agent who decides to act, remains mysterious. We can map the circuits. We can stimulate cortex and produce movement. But we can't explain why it feels like anything to will a movement, why there's an experience of agency accompanying motor commands. The machinery works. The mechanisms are understood. But as with perception, understanding the machinery doesn't explain experience. The explanatory gap persists, the hard problem lurking behind every function we describe. For now, we have motor control. Next, we need to understand how the brain changes, how it learns, how experiences leave lasting traces in neural circuits. That's where memory and plasticity come in. Memory and learning. In 1953, a 27-year-old man known in the scientific literature as HM underwent experimental surgery to treat debilitating epilepsy. Seizures had plagued him since childhood, growing progressively worse until they occurred multiple times daily, rendering him unable to work or live independently. Medication had failed. Surgery was a last resort. The surgeon, William Scoville, removed tissue from both sides of HM's medial temporal lobes, including most of his hippocampus, attempting to eliminate the seizure focus. The surgery succeeded in reducing seizures, but it produced an unexpected and devastating side effect. HM could no longer form new long-term memories. He could remember his childhood, recall events from years before the surgery, hold information in mind for a few seconds or minutes. But nothing new stuck. He couldn't remember what he'd eaten for breakfast, whether he'd met you before, what had happened yesterday. Every experience evaporated within minutes, leaving no trace. He lived in a perpetual present, unable to learn new facts, remember new people, or create new autobiographical memories. Yet HM was not completely unable to learn. If you taught him a motor skill, having him trace a pattern while looking at his hand in a mirror, a difficult task that improves with practice, he got better over days of training. Each day, he'd claim never to have done the task before, insisting he had no memory of previous sessions. But his performance improved. His hands knew what to do, even though his conscious mind remembered nothing. This dissociation revealed something fundamental. Memory is not unitary. There are multiple memory systems operating through different neural mechanisms, producing different types of knowledge. The distinction that became clear through HM and similar patients is between declarative memory, conscious recollection of facts and events, and procedural memory, unconscious knowledge of skills and habits. HM had lost declarative memory but retained procedural learning. He couldn't remember doing the mirror tracing task, but his motor system learned it nonetheless. Declarative memory itself divides into episodic memory, recollection of specific events from your life, and semantic memory, knowledge of facts about the world. HM couldn't form new episodic memories, couldn't remember experiences after his surgery. His semantic memory for new facts was also impaired, though not as completely as episodic memory. He learned a few new words that entered the language after 1953, though far fewer than someone with intact memory would acquire. The hippocampus, the structure removed from HM's brain, is essential for consolidating new declarative memories. It's not where memories are permanently stored, but it's necessary for transferring information from short-term to long-term storage. Damage the hippocampus, and new information can't be consolidated. Old memories, formed before the damage, typically remain intact, suggesting they're stored elsewhere, probably distributed across the cortex. But how does the brain actually store memories? What physical change in neural tissue corresponds to a memory? The answer, worked out over decades of research, is synaptic plasticity. Memories are encoded by changes in the strength of synaptic connections between neurons. When you learn something, specific synapses strengthen or weaken, altering how information flows through neural circuits. The pattern of synaptic strengths across billions of synapses encodes your knowledge and experiences. This idea traces back to Donald Hebb, a Canadian psychologist who in 1949 proposed what's now called Hebbian learning. Neurons that fire together wire together. If two neurons are repeatedly active at the same time, the synapse between them strengthens. This makes it more likely that activity in one will trigger activity in the other in the future. The mechanism underlying Hebbian plasticity is called long-term potentiation, LTP, discovered in the hippocampus in the 1970s. When a presynaptic neuron fires repeatedly while the postsynaptic neuron is depolarized, the synapse strengthens, becoming more effective at transmitting signals. This strengthening can last for hours, days, or longer. LTP involves several mechanisms. In the short term, existing receptors on the postsynaptic membrane become more responsive. More channels open. The synapse transmits signals more effectively. Over longer timescales, new receptors are inserted into the membrane. The postsynaptic density, the protein structure at the synapse, enlarges. The synapse physically grows. With sustained activity, entirely new synapses can form, creating additional connections between neurons. The opposite process, long-term depression, LTD, weakens synapses. If a presynaptic neuron fires while the postsynaptic neuron remains inactive, the synapse becomes less effective. Receptors are removed. The synapse shrinks. This pruning is as important as strengthening. Memory isn't just about forming new connections, but also eliminating irrelevant ones, refining circuits to encode specific patterns. The most studied mechanism involves NMDA receptors, a type of glutamate receptor with unusual properties. DA receptors act as coincidence detectors. They open only when two conditions are met simultaneously. Glutamate is bound to the receptor, and the postsynaptic membrane is depolarized. This means they detect correlated activity between presynaptic and postsynaptic neurons, exactly the condition Hebb proposed for learning. When NMDA receptors open, calcium flows into the postsynaptic neuron. This calcium influx triggers a cascade of molecular events that ultimately strengthen the synapse. Calcium activates enzymes, particularly calcium calmodulin-dependent kinase IODHAR, CAM-KII, which phosphorylates other proteins, altering their function. These changes increase the number and effectiveness of AMPA receptors, another type of glutamate receptor that mediates normal synaptic transmission. This is the molecular machinery of learning. Experience drives neural activity. Correlated activity opens NMDA receptors. Calcium floods in. Molecular cascades are triggered. Synapses change. The circuit is rewired, encoding information about the experience. But synaptic plasticity alone doesn't fully explain memory. There's also systems consolidation, the process by which memories gradually transfer from hippocampus to cortex. Initially, when you learn something new, the memory depends on the hippocampus. Damage it soon after learning, and the memory is lost. But over time, days to years, the memory becomes less dependent on the hippocampus and more distributed across cortical regions. This consolidation likely occurs during sleep. While you sleep, particularly during slow-wave sleep, the hippocampus replays patterns of activity from the day, reactivating recently formed memories. These reactivations drive plasticity in cortical circuits, gradually strengthening cortical representations until they can support the memory independently of the hippocampus. This is why sleep is crucial for memory. Sleep deprivation impairs consolidation. Students who study all night before an exam might retain information briefly but forget it rapidly. Those who study and then sleep consolidate the information more effectively, retaining it long term. There's also evidence that memories are reconsolidated each time they're retrieved. When you recall a memory, it becomes temporarily labile, susceptible to modification before being stored again. This is why memories aren't perfect recordings. Each retrieval is a reconstruction, potentially introducing errors. Memories can be distorted by suggestion, merged with other memories, even entirely fabricated, all while feeling completely genuine. Elizabeth Loftus demonstrated this in the 1970s with studies showing how easily false memories can be implanted. Show people a video of a car accident and then ask, how fast were the cars going when they smashed into each other? Versus, how fast were they going when they hit each other? The word choice influences their memory. Those who heard smashed recall higher speeds and are more likely to falsely remember broken glass that wasn't present. Memory is not a video recorder. It's a reconstruction, influenced by expectations, suggestions, emotions, and subsequent experiences. This has profound implications for eyewitness testimony in legal settings, for recovered memories in therapy, for any situation where memory accuracy matters. Confidence in a memory doesn't guarantee accuracy. People can be entirely certain of memories that are demonstrably false. Working memory is another form, distinct from long-term memory. It's the ability to hold information in mind temporarily, for seconds to minutes, while actively using it. When you mentally calculate a tip, remember a phone number long enough to dial it. Follow a conversation. You're using working memory. Working memory has limited capacity, famously described as 7 plus or minus 2 chunks of information. You can hold about that many items in mind simultaneously. Try to hold more, and earlier items start dropping out. This limitation is why you can't easily multiply large numbers in your head or remember long lists without rehearsal or chunking, grouping items into meaningful units. The prefrontal cortex is critical for working memory. Neurons here maintain sustained activity during delay periods, holding information online. Damage to prefrontal cortex impairs working memory, making it difficult to keep goals in mind, follow complex instructions, or resist distraction. Working memory is also where executive functions operate: planning, inhibition, cognitive flexibility, the ability to manipulate information mentally. These functions depend on the prefrontal cortex and mature slowly, not reaching full development until the mid-20s. This is why children and adolescents struggle with impulse control and long-term planning. Their prefrontal cortex is still developing. Then there's emotional memory, processed partly through the amygdala. Emotional events are remembered more vividly than neutral ones. The amygdala modulates memory consolidation in the hippocampus, strengthening memories for emotionally significant experiences. This is why you remember where you were during shocking events, flashbulb memories, with unusual clarity, though research shows even these memories aren't as accurate as they feel. Stress hormones, particularly cortisol, affect memory formation. Moderate stress enhances consolidation. This makes evolutionary sense. Emotionally arousing events are often important, worth remembering. But chronic stress or extreme acute stress impairs memory. Cortisol can damage the hippocampus with prolonged exposure, which may contribute to memory problems in people with PTSD or chronic anxiety. Memory also changes across the lifespan. Childhood amnesia, the inability to remember events from early life, reflects the immaturity of the hippocampus and prefrontal cortex in young children. Most people's earliest memories date to around age 3 or 4, though some recall earlier events. In aging, memory declines, particularly episodic memory and processing speed. Semantic memory, knowledge of facts, often remains stable or even improves with age as more information accumulates. The distinction between normal age-related decline and pathological conditions like Alzheimer's disease can be subtle initially, but becomes clear as pathology progresses. Alzheimer's begins in the hippocampus and adjacent regions, producing memory loss as the first prominent symptom. As the disease spreads through cortex, other cognitive functions deteriorate. Language, spatial navigation, executive functions, eventually personality and basic self-care abilities are lost. The pathology involves abnormal protein deposits, amyloid plaques, and tau tangles that disrupt neural function and trigger cell death, But here's what remains puzzling about memory. We can describe synaptic plasticity in molecular detail. We can observe LTP and LTD. We can identify the brain regions involved in different memory systems. We can even predict, to some extent, which experiences will be remembered based on neural activity patterns during encoding. What we can't explain is the subjective experience of remembering. Why does retrieving a memory feel like anything? When you recall your childhood home, you might have a visual image, a sense of the layout, emotional associations, perhaps even smells or sounds. This experience, the quale of memory, the what it's like to remember, is not explained by synaptic plasticity. The pattern of synaptic strengths encodes information. Reactivating that pattern retrieves the information. But why does retrieval produce conscious experience? Why is there something it's like to remember rather than the information simply being available for use, influencing behavior without generating subjective experience? This is the hard problem again, appearing in every domain we examine. Synaptic plasticity explains how information is stored and retrieved. It doesn't explain why retrieval feels like remembering, why there's an experience accompanying the process. Memory demonstrates the brain's remarkable ability to change, to be shaped by experience, to encode information in its physical structure. The mechanisms are understood in impressive detail. But, as with perception and action, understanding the mechanisms leaves the deepest question unanswered. Still, what we do understand is extraordinary. Your experiences literally rewire your brain. Every conversation, every skill practiced, every fact learned, alters synaptic connections. You are, in a very real sense, the sum of your memories. Your knowledge encoded in patterns of synaptic strengths distributed across billions of connections. And those patterns are constantly changing, updated by new experiences, refined by retrieval, consolidated during sleep. The machinery of memory is physical, molecular, electrical. But it produces something that feels profoundly non-physical. The continuity of self. The sense that you're the same person who existed yesterday. The ability to learn from the past and plan for the future. All of it resting on synapses, changing strength, one experience at a time. Emotion and motivation. In 1848, a railroad construction foreman named Phineas Gage was preparing a blasting charge when something went catastrophically wrong. The explosive detonated prematurely, driving a 3-foot iron tamping rod through his left cheek, behind his eye, through the front of his brain, and out the top of his skull. Remarkably, Gage survived. He was conscious within minutes, able to walk and speak. Physically, he recovered well enough to live another 12 years. But according to his physician, Dr. John Harlow, something fundamental had changed. Before the accident, Gage had been responsible, hardworking, well-liked. After, he became impulsive, profane, unable to plan or follow through on decisions. His friends said he was no longer Gage. The iron rod had destroyed much of his prefrontal cortex, particularly the orbitofrontal and ventromedial regions. For over a century, Gage's case has been cited as evidence that the frontal lobes are critical for personality, decision-making, and emotional regulation. Though recent scholarship suggests the behavioral changes were exaggerated in retellings, there's no doubt that prefrontal damage profoundly affects emotional and social functioning. Modern patients with similar damage show a consistent pattern. They're not paralyzed or intellectually impaired in obvious ways. IQ tests often return normal scores, but their lives fall apart. They make disastrous decisions, particularly in social and financial domains. They seem unable to learn from mistakes, repeating the same poor choices despite negative consequences. Marriages fail, jobs are lost. They alienate friends and family. What's wrong? The answer reveals something crucial about how emotion and cognition interact, about how the brain makes decisions. Antonio Damasio, a neurologist who studied patients with ventromedial prefrontal damage extensively, proposed that these patients lack normal emotional responses to anticipated outcomes. When faced with a decision, they can reason through options logically, list pros and cons, understand intellectually what might happen. But they don't feel anything about those outcomes. Without emotional signals, what economists call somatic markers, they can't assign value to different options. All choices feel equivalent. Damasio tested this with a gambling task. Patients drew cards from different decks, winning or losing money. Some decks offered occasional large rewards, but frequent larger losses, net negative. Other decks offered smaller rewards, but infrequent small losses, net positive. Normal subjects quickly learned to avoid the bad decks, even before they could articulate why. They developed anticipatory stress responses measured by skin conductance when reaching for bad decks. Patients with ventromedial prefrontal damage never developed these emotional responses. They understood intellectually when asked which decks were better, but without the gut-level feeling. The emotional signal warning them away from bad choices, they continued selecting from losing decks. Emotion wasn't interfering with rational decision-making. It was necessary for it. This overturns centuries of philosophical tradition that treated emotion and reason as opposed, with reason as the superior faculty that should override emotion. The brain doesn't work that way. Emotion and cognition are deeply integrated. You can't make good decisions with reason alone. Emotion is generated through interconnected systems, particularly the limbic system and its connections with prefrontal cortex. The amygdala, which we've mentioned before, is central to fear and threat detection. It receives sensory information directly from the thalamus, bypassing cortex, allowing rapid emotional responses before conscious processing occurs. This is the low road to emotion, fast but crude. You jump at a shape that might be a snake before consciously recognizing it's a stick. Better safe than sorry from an evolutionary perspective. The amygdala also receives information from cortex, the high road, allowing more nuanced emotional responses based on context and prior knowledge. The amygdala projects widely throughout the brain, influencing perception, attention, memory, and action. When you're afraid, the amygdala enhances processing of threat-related information, biases attention toward potential dangers, strengthens memory for fearful events, and prepares the body for action through connections to the hypothalamus and brainstem. The hypothalamus orchestrates physiological responses to emotion through the autonomic nervous system and endocrine system. Fear activates the sympathetic nervous system. Heart rate increases, blood pressure rises, pupils dilate, digestion slows, stress hormones flood the bloodstream. The body prepares for fight or flight. The hypothalamus also controls the HPA axis, hypothalamic pituitary adrenal axis, a hormonal cascade that produces cortisol. When you experience stress, the hypothalamus releases corticotropin-releasing hormone, CRH, which triggers the pituitary gland to release adrenocorticotropic hormone, ACTH, which signals the adrenal glands to produce cortisol. This whole cascade takes minutes, slower than the immediate sympathetic response but with longer-lasting effects. Cortisol mobilizes energy, suppresses non-essential functions like digestion and reproduction, and modulates immune responses. In acute stress, this is adaptive. It helps you deal with immediate threats. But chronic stress with persistently elevated cortisol damages health. It impairs immune function, promotes fat storage particularly around the abdomen, increases cardiovascular disease risk, and can damage the hippocampus, impairing memory. The ventromedial prefrontal cortex, the region damaged in Phineas Gage, regulates emotional responses through top-down control. It doesn't eliminate emotion but modulates it, contextualizing emotional reactions, inhibiting inappropriate responses, selecting context-appropriate behavior. Damage here doesn't eliminate emotion. Patients still feel. But the regulation is impaired, leading to emotional responses that don't match situations. Other prefrontal regions contribute to emotional regulation through reappraisal, the ability to reinterpret situations to change emotional responses. If you're anxious about a presentation, you might reframe it as an opportunity rather than a threat. This cognitive reappraisal activates dorsolateral prefrontal cortex and reduces amygdala activity, literally dampening the emotional response through deliberate reinterpretation. Reward processing involves different circuits, particularly the ventral tegmental area, VTA, and nucleus accumbens, along with their connections to prefrontal cortex. These structures use dopamine as their primary neurotransmitter. When you experience something rewarding, food, sex, social approval, unexpected good fortune, dopamine neurons in the VTA fire, releasing dopamine in the nucleus accumbens and prefrontal cortex. For decades, dopamine was thought to encode pleasure, the subjective enjoyment of rewards. But research has complicated this picture. Dopamine seems to encode prediction error, the difference between expected and received reward. When something better than expected happens, dopamine neurons fire strongly. When reward matches expectations, they fire moderately. When reward is worse than expected, they decrease firing below baseline. This signal teaches the brain which actions lead to rewards, enabling learning. The dopamine prediction error adjusts synaptic strengths in circuits involved in selecting actions, gradually biasing behavior toward rewarding outcomes. This is reinforcement learning, implemented neurally through dopamine-modulated plasticity. Addictive drugs hijack this system. Cocaine, amphetamines, and other stimulants increase dopamine levels directly, producing euphoria and powerful reinforcement. The brain learns that drug taking is extraordinarily rewarding, even as tolerance develops and the actual pleasure diminishes. The learning persists, driving compulsive drug seeking even when it no longer produces enjoyment, even when consequences are devastating. This dissociation between wanting and liking, between motivation and pleasure, reveals that reward involves multiple components. Dopamine drives wanting, the motivation to pursue rewards. but liking, the actual subjective pleasure, involves different systems, particularly opioid and cannabinoid signaling in the nucleus accumbens and elsewhere. You can want something intensely without liking it much, as occurs in addiction. Depression involves dysregulation of emotional systems. It's not simply sadness, it's anhedonia, inability to experience pleasure, along with negative emotional bias, where attention and memory preferentially process negative information. Antidepressants, particularly SSRIs, increase serotonin levels, but how this alleviates depression isn't fully understood. Serotonin affects mood, but the mechanisms linking serotonergic signaling to subjective emotional experience remain unclear. The insula, a cortical region folded deep within the lateral sulcus, integrates bodily sensations with emotional experience. It processes interoception, awareness of internal bodily states, heartbeat, breathing, gut sensations. The insula helps generate the feeling component of emotion, the subjective experience of feeling afraid, disgusted, happy. Disgust particularly involves the insula. Seeing something disgusting, rotting food, bodily waste, activates the insula, producing both the feeling of disgust and the characteristic facial expression and nausea. Social and moral disgust, feeling repelled by immoral behavior, also activates the insula, suggesting that moral emotions might have evolved from more basic disgust responses to physical contaminants. The anterior cingulate cortex, wrapping around the corpus callosum, is involved in conflict monitoring and emotional regulation. It detects situations where multiple response options compete, signaling the need for increased cognitive control. It's also involved in processing social pain, the distress of rejection or exclusion, which activates overlapping circuits with physical pain. This neural overlap between social and physical pain isn't metaphorical. Social rejection literally hurts, activating pain processing regions. Acetaminophen, a physical pain reliever, reduces reports of hurt feelings in some studies. A bizarre result suggesting that social and physical pain share mechanisms. Emotion also has a strong social component. The ability to recognize emotions in others through facial expressions, tone of voice, body language is critical for social interaction. The fusiform face area in the temporal lobe specializes in face recognition. Adjacent regions process facial expressions, extracting emotional information. Mirror neurons discovered in monkey motor cortex but also found in humans fire both when performing an action and when observing someone else perform it. This has led to speculation that mirror neurons underlie empathy, the ability to understand others' emotions by simulating them internally. The evidence remains debated, but there's no doubt that observing others' emotions activates similar neural circuits to experiencing those emotions oneself. Empathy involves multiple components. Emotional contagion, automatically feeling what others feel, is rapid and unconscious. Cognitive empathy, understanding intellectually what someone feels, involves perspective-taking and theory of mind. Empathic concern, feeling compassion and motivation to help, involves additional circuits in prefrontal cortex and anterior cingulate. Psychopathy, characterized by lack of empathy and remorse, involves abnormalities in the amygdala and ventromedial prefrontal cortex. Psychopaths can understand intellectually what others feel, but don't experience the emotional resonance that typically accompanies this understanding. They recognize fear in others, but aren't moved by it. Motivation is closely tied to emotion through reward and aversion systems. Goals are pursued because achieving them is rewarding, or because not achieving them is aversive. The balance between approach motivation, seeking rewards, and avoidance motivation, escaping threats, varies across individuals and contexts. Prefrontal cortex represents goals and maintains them over time despite distractions and obstacles. Damage here produces goal neglect, where people understand what they should do but fail to organize behavior around long-term objectives. They become stimulus-driven, responding to immediate environmental cues rather than internal goals. But here's the puzzle emotion presents for neuroscience. We can map the circuits involved in different emotions. We can identify the neurotransmitters and hormones. We can measure physiological responses and observe behavioral changes. We can even induce emotions through electrical stimulation of specific brain regions. What we can't explain is why any of this feels like anything. Why does amygdala activation produce the subjective experience of fear? Why do we feel sad when serotonin signaling is disrupted? The neural correlates of emotion are well documented. The mechanism by which neural activity becomes subjective feeling is not. This is, once again, the hard problem. We can describe the machinery of emotion in impressive detail. But describing the machinery doesn't explain qualia, the subjective qualities of emotional experience. Fear feels a certain way. Joy feels different. These are not simply different patterns of neural activity, though they certainly involve different patterns. They're experiences, felt from the inside, and we don't know how neural activity produces them. Emotion reveals the integration of brain and body, cognition and feeling, reason and passion. It demonstrates that the brain doesn't just process information coldly, but assigns value, generates motivation, creates the subjective sense of caring about outcomes. Without emotion, decisions become impossible. Without emotional memory, learning is impaired. Without social emotions, cooperation collapses. The machinery is physical, cellular, chemical, but it produces something that feels deeply personal and subjective. Your emotional life, the feelings that color your experience, motivate your actions, define your relationships, and make existence meaningful rather than merely mechanical. Consciousness and higher functions. We've mapped the brain's territories, traced how neurons communicate, examined sensory processing, motor control, memory, and emotion. We've described mechanisms in molecular detail, but we've been circling around something without confronting it directly. Consciousness. You are conscious right now. You're aware of these words, experiencing thoughts about them, perhaps aware of sounds around you, sensations from your body, the continuous feeling of being present. There is something it's like to be you, a subjective perspective, an inner experience that accompanies your neural activity. This is the most obvious fact about your mental life and, simultaneously, the most mysterious. Every other brain function we've discussed, perception, movement, memory, emotion, can be described in terms of information processing, neural circuits performing computations. But consciousness seems different. It's not just processing information, it's experiencing it. The hard problem of consciousness, articulated most clearly by philosopher David Chalmers in the 1990s, is explaining why there's subjective experience at all. Why doesn't information processing happen in the dark? influencing behavior without generating any inner experience. Why is there something it's like to see red, to feel pain, to taste coffee? Neuroscience can identify neural correlates of consciousness, patterns of brain activity that correspond to conscious states. When you're conscious, certain patterns appear. When you're unconscious, under anesthesia or in dreamless sleep, those patterns disappear. We can even manipulate consciousness, inducing or suppressing it through drugs, brain stimulation, or damage. But correlation isn't explanation. Finding neural correlates doesn't explain why those particular patterns produce experience. It doesn't bridge the gap between objective, third-person descriptions of neural activity and subjective, first-person experience. Some neuroscientists believe the hard problem will eventually be solved through continued investigation of neural mechanisms. Others think it's fundamentally intractable, that consciousness might be forever mysterious. Still others argue the problem is misconceived, that we're asking the wrong questions or applying concepts inappropriately. Setting aside the hard problem, there are tractable questions about consciousness that neuroscience has made progress on. What are the neural conditions necessary for consciousness? What functions does consciousness serve? How does the conscious brain differ from the unconscious brain? The cerebral cortex is necessary for consciousness, at least the kind of consciousness humans experience. Damage to the cortex can eliminate or severely alter consciousness. Decerebrate animals with cortex removed but brainstem intact show sleep-wake cycles and basic reflexes but appear to lack subjective experience. The brainstem maintains arousal, but consciousness requires cortex. Within the cortex, certain regions seem more important than others. The posterior cortex, parietal and occipital lobes particularly, generates the content of consciousness, what you're currently aware of. Visual consciousness depends on visual cortex. Conscious awareness of bodily sensations depends on somatosensory cortex. The prefrontal cortex contributes differently. It's not where conscious content arises, but where information becomes accessible to work in memory, available for report, decision-making, and deliberate control. You can process information in posterior cortex without being consciously aware of it, as demonstrated by blindsight. But for information to enter working memory, to become something you can talk about and use flexibly, prefrontal involvement is necessary. There's also the thalamus, which regulates cortical activity and serves as a hub connecting different cortical regions. Damage to certain thalamic nuclei can cause coma or persistent vegetative state. The thalamus and cortex form reciprocal loops, with information cycling between them, and this thalamocortical system seems essential for generating conscious states. One influential theory proposed by Giulio Tononi is Integrated Information Theory, IIT. This theory suggests that consciousness corresponds to integrated information that is both differentiated, representing many possible states, and unified, forming a single interconnected system rather than independent subsystems. IIT proposes a measure called PHI, representing the degree of integration. Systems with high phi, lots of differentiated states that are tightly integrated, have consciousness. Systems with low phi, either because they're simple or because they're modular with parts that don't interact, lack consciousness. The theory makes testable predictions. The cerebral cortex, highly interconnected with complex dynamics, should have high phi. The cerebellum, despite containing more neurons than cortex, is more modular with less integration between regions, so should have lower phi and not contribute to consciousness. Clinical evidence supports this. Cerebellar damage impairs motor control, but doesn't eliminate consciousness. Cortical damage can. Another theory, global workspace theory proposed by Bernard Bars and developed computationally by Stanislas Dehaene, suggests that consciousness arises when information becomes globally available to multiple cognitive systems. Unconscious processing is local, confined to specific modules. Conscious processing involves broadcasting information across a global workspace, making it accessible to work in memory, attention, language, and decision-making systems. On this view, consciousness is like a spotlight. Vast amounts of processing occur unconsciously in the dark. Consciousness illuminates a small subset, bringing it into the global workspace where it can be reported and used flexibly. This explains why consciousness has limited capacity. The global workspace is a bottleneck. Only a small amount of information can be broadcast widely at any moment. Both theories have empirical support and critics. Neither fully solves the hard problem. They describe conditions under which consciousness arises, but don't explain why those conditions produce subjective experience. Still, they offer frameworks for investigating consciousness scientifically, making predictions that can be tested. The prefrontal cortex also supports metacognition, thinking about thinking. You're not just conscious of the world, but conscious of being conscious. You can reflect on your thoughts, monitor your knowledge, assess your confidence. This self-awareness, this recursive quality of consciousness, involves prefrontal cortex and its connections to other regions. Damage to prefrontal cortex impairs metacognitive abilities. Patients may have conscious experiences, but struggle to monitor or report on them accurately. They're less aware of their own knowledge gaps, less able to adjust confidence based on evidence, less capable of the kind of introspective awareness that characterizes normal consciousness. The sense of self, the feeling that there's a unified I experiencing thoughts and perceptions, is constructed by the brain. This might seem obvious, but it has surprising implications. The self isn't a thing or place in the brain, but a process, a pattern of neural activity that creates the illusion of a unified observer. This illusion can be disrupted. In split-brain patients, where the corpus callosum connecting the hemispheres is severed to treat epilepsy, each hemisphere can process information independently. Present a word to the left visual field, processed by the right hemisphere, and patients can't verbally report it because language is typically in the left hemisphere. But they can point to the corresponding object with their left hand, controlled by the right hemisphere. Each hemisphere seems to have its own awareness, its own perspective. More dramatically, certain neurological conditions produce alterations in self-experience. Out-of-body experiences, where people feel they're observing themselves from outside their body, can be triggered by stimulation of the temporoparietal junction. The sense of body ownership, the feeling that your limbs belong to you, can be manipulated with clever setups like the rubber hand illusion, where stroking a rubber hand while stroking your hidden real hand creates the sensation that the rubber hand is yours. These manipulations reveal that the sense of self is constructed from multisensory integration. The brain combines visual, tactile, proprioceptive information to create a coherent sense of body ownership and spatial location. Disrupt that integration, and the sense of self fragments or distorts. Executive functions, higher-order cognitive processes like planning, decision-making, cognitive flexibility, and inhibitory control, depend heavily on prefrontal cortex. These are the functions that allow you to pursue long-term goals, resist immediate temptations, switch between tasks, and override habitual responses. Work-in memory, which we touched on earlier, is fundamental to executive function. It allows you to hold information in mind while manipulating it. Solve a math problem mentally, follow a conversation, navigate to a destination, all require working memory. The dorsolateral prefrontal cortex is particularly critical, maintaining information active during delays and protecting it from interference. Cognitive control. The ability to regulate thoughts and actions involves detecting conflict between response options and adjusting behavior accordingly. The anterior cingulate cortex monitors for conflict, signaling when control is needed. The dorsolateral prefrontal cortex then implements control, biasing processing toward task-relevant information and suppressing distractions. Language is another uniquely human higher function. While many animals communicate, human language is qualitatively different in its complexity, flexibility, and generative power. From a finite set of words and grammatical rules, we can produce and understand an infinite number of sentences, expressing abstract thoughts, discussing past and future, creating fiction. Language depends primarily on the left hemisphere in most people. Broca's area in the left frontal lobe is critical for speech production. Damage here produces expressive aphasia, where comprehension remains intact, but speech becomes effortful, halting, ungrammatical. Patients know what they want to say, but struggle to produce the words. Wernicke's area in the left temporal lobe is critical for comprehension. Damage produces receptive aphasia, where speech is fluent and grammatical but meaningless. And comprehension is severely impaired. Patients speak nonsensically, substituting incorrect words without recognizing the errors, and cannot understand spoken or written language. These areas are connected by the arcuate fasciculus, a white matter tract. Damage to this connection produces conduction aphasia, where both production and comprehension are relatively preserved. But patients cannot repeat sentences they've just heard, because the connection between comprehension and production systems is disrupted. Language also interacts profoundly with thought. The Sapir-Whorf hypothesis suggests that language shapes thought, that speakers of different languages conceptualize the world differently because of linguistic differences. The strong version of this hypothesis, linguistic determinism, is largely discredited. But there's evidence for a weaker version, linguistic relativity, where language influences thought without fully determining it. For instance, languages differ in how they encode spatial relationships, time, color, causality. These differences correlate with how speakers of those languages perform on non-linguistic cognitive tasks, suggesting language influences, though doesn't completely determine thought. Creativity, The ability to generate novel and valuable ideas involves widespread brain networks. The default mode network, active during rest and mind wandering, appears important for creative thinking, generating unconstrained associations and exploring possibilities. The executive control network, including prefrontal cortex, then evaluates and refines these ideas. Creative insights often occur during moments of relaxed attention, when the default mode network is active and constraints are loosened. This is why people report having insights in the shower or while walking, activities that occupy enough attention to prevent focused thought, but not so much that the mind can't wander. Theory of mind, the ability to attribute mental states to others, to understand that other people have beliefs, desires, intentions that differ from your own, is critical for social cognition. It emerges in children around age 4, when they pass the false belief task, understanding that someone can hold a belief they know to be false. Theory of mind involves the medial prefrontal cortex, temporoparietal junction, and superior temporal sulcus. Damage to these areas or developmental differences, as in autism spectrum disorder, impair the ability to intuit others' mental states, making social interaction challenging. All of these higher functions, consciousness, metacognition, executive control, language, creativity, theory of mind, emerge from neural mechanisms. They're products of the same electrochemical machinery that processes sensory input and controls movement. Neurons firing in patterns, synapses transmitting signals, circuits computing over distributed networks. Yet somehow, from this machinery, the uniquely human mind emerges. The capacity for abstract thought, moral reasoning, artistic creation, scientific discovery, the ability to reflect on our own existence, to ask why we're conscious, to build models of reality and test them against experience. We can describe the neural substrates of these functions. We can identify necessary and sufficient conditions. We can manipulate them and observe the consequences. But we cannot yet explain, in any satisfying way, how neurons become thoughts, how patterns of activity become the felt quality of experience, how physical processes produce the mental realm. The machinery is physical. The experience is something else. Not non-physical, necessarily, but subjective, private, accessible only from the first-person perspective. And that gap between objective description and subjective experience, between what can be measured and what can only be felt, remains the deepest mystery in neuroscience. Perhaps future discoveries will bridge this gap. Perhaps we'll find that consciousness, like life before molecular biology, is simply a complex organization of ordinary physical processes, mysterious only until we understand the details. Or perhaps consciousness points to something genuinely new, an emergent property that cannot be reduced to or predicted from its components. For now, we have the hardware and increasingly detailed understanding of how it works. What we're still missing is the full explanation of why working hardware produces the software of consciousness, the felt experience of being a mind. Brain development and plasticity. The human brain begins as a small sheet of cells, barely distinguishable from the tissue that will become skin or gut. 3 weeks after conception, this sheet folds inward, forming a hollow tube called the neural tube. From this simple beginning, through a precisely orchestrated series of events, emerges the most complex structure in the known universe. What's remarkable about brain development is not just the final result, but how it gets there. The adult brain contains roughly 86 billion neurons and perhaps 100 trillion synaptic connections. None of this is directly encoded in your DNA. You have about 20,000 genes total, nowhere near enough to specify every connection, every circuit. The brain must assemble itself through a combination of genetic instruction and experience-dependent refinement. Neural development follows several stages, each with its own mechanisms and vulnerabilities. The process begins with neurogenesis, the birth of new neurons. In the developing fetus, neural stem cells in the ventricular zone, lining the fluid-filled spaces inside the brain, divide rapidly. Some divisions produce more stem cells, maintaining the pool. Others produce neurons that will migrate to their final destinations. Neuronal migration is a journey. Neurons don't simply appear where they're needed. They are born in one location and must travel, sometimes millimeters, an enormous distance at cellular scale, to reach their proper position. They follow chemical gradients and physical guides, radioglial cells that extend from the ventricular zone to the brain's surface, forming scaffolding along which neurons climb. This migration is vulnerable to disruption. Alcohol exposure during pregnancy, certain genetic mutations, infections, all can interfere with neuronal migration, producing disorders where neurons end up in wrong locations. Lissencephaly, smooth brain, results from migration failure, leaving the cortex unfolded and severely underdeveloped. Affected individuals have profound intellectual disability and seizures. The brain's architecture depends on neurons reaching their correct destinations. Once neurons arrive, they extend axons and dendrites, seeking connection partners. A growing axon has a specialized tip called a growth cone, studded with receptors that detect chemical signals in the environment. Some chemicals attract the growth cone, others repel it. The axon navigates through the developing brain like a ship using lighthouses and hazard markers, following gradients of guidance molecules to reach its target. Finding the right target region isn't enough. The axon must then find the right type of cell, sometimes the right individual cell within that region. This requires additional molecular matching. Cells display identity tags on their surface, combinations of proteins that specify cell type and position. Growth cones sample these tags, forming synapses only with appropriate partners. But the initial wiring is crude. Far too many connections form, a profusion of synapses that must be refined. This is where experience enters. Neural activity, driven by spontaneous firing in the developing brain and later by sensory experience, strengthens some synapses while weakening others. The principle is Hebbian. Synapses that successfully drive postsynaptic firing are strengthened. Those that don't are weakened and eventually eliminated. This process, synaptic pruning, is as important as synapse formation. At birth, an infant has far more synapses than an adult. Throughout childhood and adolescence, synapses are eliminated, roughly half lost by adulthood. This might seem wasteful, but it's essential. The pruning refines circuits, removing incorrect connections and strengthening appropriate ones, sculpting the brain based on experience. Different brain regions mature at different rates. Sensory areas develop early. primary visual cortex, for instance, undergoes massive synapse formation and pruning in the first few years of life. This is a critical period for vision. Deprive a young animal of visual input during this window by covering one eye, and that eye's connections in the cortex never develop normally. The neurons become dominated by input from the open eye. If uncovered later, after the critical period closes, vision in the deprived eye remains permanently impaired. A condition called amblyopia. Critical periods exist for many functions. Language acquisition is most efficient before puberty. Children exposed to language from birth become native speakers with perfect grammar and pronunciation. Adults learning a second language rarely achieve native fluency, particularly in pronunciation. The brain's language circuits become progressively less plastic with age. This doesn't mean adults can't learn. Adult brains retain substantial plasticity, but learning mechanisms shift from the exuberant, experience-expectant plasticity of childhood to more constrained, experience-dependent plasticity in adulthood. Children's brains are optimized for rapid learning across domains. Adult brains are more specialized, with learning focused within established frameworks. The prefrontal cortex is the last region to fully mature, not reaching adult organization until the mid-20s. This protracted development explains adolescent behavior. The limbic system, including the nucleus accumbens and amygdala, matures earlier, driving reward-seeking and emotional reactivity. But the prefrontal control systems that inhibit impulses and weigh long-term consequences are still developing. This creates an imbalance. Adolescents are sensitive to rewards, peer influence, and immediate gratification, but their ability to control impulses and consider future consequences is still maturing. This isn't a character flaw, it's neurodevelopment. The systems don't mature at the same rate, producing the characteristic decision-making patterns of adolescence. Myelination, the process of wrapping axons in insulating myelin, also continues into the 20s. White matter tracks, the connections between brain regions, become increasingly efficient as myelination proceeds. This improves processing speed and integration across regions, contributing to the gradual improvements in executive function and cognitive control through adolescence into early adulthood. Brain development can go wrong in numerous ways, producing developmental disorders. Autism spectrum disorder involves alterations in brain connectivity, with differences in how regions communicate. The details vary across individuals, but many show increased local connectivity, stronger connections within regions, and decreased long-range connectivity between distant regions. This affects information integration, potentially explaining some autistic traits. Enhanced attention to details, preference for predictable patterns, difficulty with flexible thinking that requires coordinating information across multiple systems, all might relate to altered connectivity patterns. The spectrum nature of autism, with enormous variation in symptoms and severity, reflects the complexity of brain development. Many routes can lead to altered connectivity. ADHD, attention deficit hyperactivity disorder, involves differences in prefrontal cortex and its connections to the basal ganglia. Executive functions, attention, and impulse control, all prefrontal dependent, are impaired. The disorder often improves with age as prefrontal cortex matures, though many adults retain some symptoms. Schizophrenia typically emerges in late adolescence or early adulthood, despite being a neurodevelopmental disorder with genetic basis. Current theories suggest that excessive synaptic pruning during adolescence might contribute. The normal refinement process goes too far, eliminating too many connections, particularly in prefrontal cortex. This produces the cognitive and social impairments characteristic of schizophrenia. But the brain's capacity for change doesn't end with development. Adult plasticity remains substantial, enabling learning throughout life. Every skill you acquire, every fact you learn, changes your brain. London taxi drivers, who must memorize the city's complex layout, show enlarged hippocampi compared to controls. Musicians who train extensively show enlarged auditory and motor cortices in regions representing the hands and auditory processing. These aren't trivial changes. They're structural alterations visible on brain scans. Experience literally reshapes the brain, adjusting the volume of different regions, the density of connections, the efficiency of circuits. This is neuroplasticity in action. Recovery from brain injury demonstrates plasticity dramatically. After stroke, which kills neurons by depriving them of oxygen, lost functions can sometimes be regained through rehabilitation. The damaged tissue doesn't regenerate. Instead, surviving regions take over functions of damaged areas. This functional reorganization has limits. It works better for some functions than others, better in younger patients than older, better with smaller lesions than larger. But the fact that it happens at all demonstrates the brain's capacity for reorganization. Physical rehabilitation exploits plasticity. Repeated practice of impaired movements drives plasticity in motor cortex and related areas. Constraint-induced movement therapy, where the functional limb is restrained, forcing use of the impaired limb, produces better recovery than conventional therapy, precisely because it drives intense, focused practice that promotes plasticity. Even more dramatically, sensory substitution demonstrates plasticity. Blind individuals can learn to navigate using tongue-based vision systems, where a camera converts images to tactile patterns on the tongue. With practice, users report experiencing these signals as visual, not tactile. The brain has interpreted the patterns, originally processed in somatosensory cortex, as visual information. Congenitally blind individuals show reorganization where visual cortex, deprived of visual input, becomes recruited for auditory and tactile processing. This is called cross-modal plasticity. The cortex doesn't sit idle. It processes whatever input is available. Blind individuals who read Braille show activation of visual cortex while reading, suggesting this cortex has been repurposed for tactile processing. Meditation provides another example of experience-dependent plasticity. Long-term meditators show structural differences in prefrontal cortex, insula, and hippocampus, regions involved in attention, interoception, and emotion regulation. These aren't pre-existing differences that attracted people to meditation, but results of practice, demonstrated by studies showing similar changes in meditation. Naive subjects after 8 weeks of training. Physical exercise also promotes plasticity, particularly in the hippocampus. Exercise increases neurogenesis, one of the few confirmed ways to generate new neurons in the adult brain. These new neurons integrate into existing circuits and contribute to memory formation. Exercise also increases brain-derived neurotrophic factor, BDNF, a protein that promotes neuronal survival and synaptic plasticity. But plasticity has a dark side. Chronic pain involves maladaptive plasticity, where pain processing circuits become hypersensitive. Phantom limb pain, where amputees feel pain in missing limbs, results from cortical reorganization following amputation. The cortical representation doesn't disappear but becomes distorted, generating pain signals from a body part that no longer exists. Addiction is another example of maladaptive plasticity. Repeated drug use changes synaptic strengths in reward circuits, creating powerful associations between drug-related cues and craving. These changes persist long after drug use stops, which is why addiction is so difficult to overcome. The brain has learned, through dopamine-mediated plasticity, that drugs are supremely important, and this learning is difficult to reverse. The capacity for plasticity raises a question about identity. If your brain is constantly changing, synapses forming and eliminating, circuits reorganizing, what maintains the continuity of self? How are you the same person you were a decade ago when the physical structure of your brain has changed substantially? There's no simple answer. Continuity of identity seems to rely on continuity of memories and personality traits, which are themselves encoded in patterns of synaptic connectivity. As long as changes are gradual, as long as new experiences build on existing structures rather than replacing them wholesale, identity persists. But dramatic brain changes can alter identity. Severe dementia, extensive brain damage, even some psychiatric conditions, can produce changes so profound that the person seems fundamentally different. They retain biological continuity, but psychological continuity is disrupted, which raises uncomfortable questions about what identity really means and where it resides. Plasticity demonstrates that the brain is not fixed hardware running software. It's hardware that rewrites itself through use. Every experience, every thought, every memory, every skill leaves physical traces. You are not separate from your brain. You are your brain, or more precisely, you are patterns of activity and connectivity in your brain. Patterns that change continuously based on experience. This has implications for education, rehabilitation, mental health treatment, and how we think about human nature. We're not born with fixed capacities that simply unfold. We're shaped by experience in profound ways. The brain develops through interaction with environment, learning through engagement with the world. This plasticity is the brain's greatest strength and a source of vulnerability. It enables adaptation, learning, recovery. But it also means early experiences shape development in lasting ways, that trauma can leave persistent marks, that skills require practice and atrophy with disuse. Understanding plasticity changes how we approach learning and brain health. Use it or lose it applies literally to neural circuits. Challenging your brain, learning new skills, maintaining physical and social activity, all promote healthy plasticity and cognitive reserve that can protect against age-related decline. The brain is not a static computer. It's a living organ that grows, adapts, reorganizes, and slowly declines. Plasticity is what makes us capable of change throughout life. And that capacity, to be shaped by experience, to learn from the world, to adapt to new circumstances, is perhaps the most remarkable feature of the most remarkable structure in the universe. The integrated brain. We've traveled through the nervous system, from its largest divisions down to individual synapses, from sensory input to motor output, from memory formation to emotional experience. We've examined consciousness, development, and plasticity. But there's a risk in this approach, the risk of seeing the brain as a collection of separate parts, rather than what it actually is, an integrated system. Nothing in the brain works alone. Vision doesn't happen only in the occipital cortex. Visual information streams forward to temporal regions for object recognition, to parietal cortex for spatial processing, to frontal cortex for planning actions. It's modulated by attention from prefrontal and parietal networks, enhanced by emotional significance through the amygdala, integrated with memory in the hippocampus. What you see is not raw sensory data, but a construction involving dozens of brain regions working together. Memory isn't stored in one place. The hippocampus is necessary for forming new declarative memories, but those memories ultimately distribute across the cortex, with different aspects in different locations. A memory of a beach might involve visual details in occipital cortex, sounds in temporal cortex, the feeling of sand in somatosensory cortex, the smell of salt air in olfactory cortex, the emotion of that moment in the amygdala and prefrontal regions. Recalling the memory reactivates all these areas in a coordinated pattern. Emotion influences everything. It modulates perception, making you more likely to notice threats when anxious, biasing attention toward emotional stimuli. It strengthens memories, particularly for emotionally significant events. It affects decision-making, providing the somatic markers that guide choices. It drives motivation, making goals feel worth pursuing. Remove emotion, as in patients with ventromedial prefrontal damage. And cognition doesn't improve. It collapses. Motor control isn't just motor cortex sending commands. It involves planning in prefrontal and parietal cortex, sequence selection in premotor areas, refinement by the cerebellum, action selection by the basal ganglia, all coordinated with sensory feedback that continuously updates and corrects ongoing movements. Every simple action, reaching for a cup, involves massive parallel processing across multiple systems. Language doesn't reside only in Broca's and Wernicke's areas. It involves auditory cortex for processing speech sounds, motor cortex for producing articulation, semantic networks distributed throughout temporal and parietal cortex for word meanings, executive systems in prefrontal cortex for constructing sentences and maintaining conversation topics. Reading recruits visual cortex. Sign language recruits motor and visual systems differently than spoken language. The brain flexibly deploys whatever circuits are available to support linguistic function. This integration is why brain damage often produces complex, multifaceted impairments rather than loss of single, isolated functions. Stroke affecting the middle cerebral artery, which supplies large portions of frontal, parietal, and temporal cortex, can simultaneously impair movement, sensation, language, attention, and spatial processing. The functions aren't separate modules that can be cleanly removed. They're aspects of an integrated system, and damage to one part disrupts many functions. The brain's integration also explains why compensatory mechanisms sometimes work. After damage to one region, other regions can partially take over its functions, not because they contain duplicate circuits, but because they're already connected to the damaged area and can be recruited to perform related computations. This is possible only because the brain is fundamentally integrated, with rich interconnections between regions. White matter, the bundles of myelinated axons connecting different brain regions, constitutes roughly half the brain's volume. This isn't empty space or mere wiring. It's infrastructure that enables integration. The corpus callosum alone, connecting the hemispheres, contains about 200 million axons. The superior longitudinal fasciculus, connecting frontal and parietal temporal regions, is equally massive. These connections make the brain more than a collection of isolated processors. They create a unified system. Information doesn't flow in one direction, from sensory input through processing stages to motor output. It flows in loops. Sensory cortex sends information forward to higher areas, but those higher areas send projections back. Feedback connections that are actually more numerous than feed-forward connections. These feedback projections allow top-down influences, expectations, and context-shaping sensory processing. This is why perception is predictive. Your brain constantly generates predictions about incoming sensory information based on context and past experience. Sensory input is then compared to predictions. Matches are processed efficiently. Mismatches, prediction errors, receive more processing. This predictive coding, as it's called, makes perception efficient, but also subject to illusions and biases. You see what you expect to see, literally, because expectations shape sensory processing. Attention is another integrative function, coordinating activity across the brain. When you attend to something, activity in regions processing that information increases, while activity in regions processing irrelevant information decreases. This isn't just one region becoming more active. It's synchronized modulation across multiple regions simultaneously. Attention involves networks spanning frontal and parietal cortex, sometimes called the dorsal attention network, that sends signals modulating sensory processing regions. These top-down signals enhance processing of attended features while suppressing distractors. Without attention, even though sensory information reaches the cortex, it doesn't fully penetrate consciousness and doesn't get encoded into memory. Attention is the gateway to awareness and learning. Sleep demonstrates brain-wide integration dramatically. During waking, the brain is in a relatively desynchronized state, different regions processing different information with different timing. During slow-wave sleep, the cortex becomes highly synchronized, neurons across vast areas firing together in slow waves, less than 1 cycle per second. This synchronization allows the hippocampus to replay recently encoded memories, reactivating cortical neurons involved in those experiences. These reactivations drive plasticity, consolidating memories from hippocampal-dependent to cortically distributed storage. Without sleep, consolidation is impaired. The integration that occurs during sleep, the coordination between hippocampus and cortex, is essential for stable long-term memory. Dreaming, which occurs primarily during REM sleep, involves a different pattern. The cortex is active, sometimes as active as during waking. But the activity is disconnected from sensory input and motor output. The brain generates experiences, often bizarre and emotionally intense, without external stimuli. Dreams demonstrate that conscious experience doesn't require sensory input. The machinery for generating experience can run offline, producing qualia without external reference. The default mode network, active during rest when you're not focused on external tasks, is another integrative system. It includes medial prefrontal cortex, posterior cingulate cortex, precuneus, and lateral parietal regions. This network activates during mind-wandering, self-referential thought, memory retrieval, imagining the future, thinking about others' mental states. The default mode network might support the sense of self, the continuous narrative you construct about your life and identity. It's where internal mentation occurs, thinking about yourself and others, simulating scenarios, reflecting on experiences. Disruption of this network, in conditions like depression or Alzheimer's disease, alters self-experience and the ability to engage in self-referential thought. The brain is also integrated with the body more thoroughly than traditionally recognized. The vagus nerve, running between brainstem and visceral organs, carries signals in both directions. It reports on heart rate, breathing, gut state, and carries commands that regulate these functions. But it also influences cognition and emotion. Stimulating the vagus nerve can reduce depression and seizures, demonstrating that the brain-body connection isn't unidirectional. The gut-brain axis, communication between the enteric nervous system in the gut and the central nervous system, influences mood, stress responses, and even cognition. The gut microbiome, bacteria residing in the intestines, affects brain function through multiple pathways, producing neurotransmitter precursors, modulating immune responses, sending signals via the vagus nerve. This is why gut health affects mental health, why antibiotics that disrupt the microbiome can temporarily alter mood. The brain is embodied, not a computer running in isolation, but an organ embedded in a body, constantly receiving signals from and sending signals to every organ system. Hormones released by endocrine glands affect neural function. The immune system communicates with the brain through cytokines, signaling molecules that produce sickness behavior when you're ill, making you lethargic, withdrawn, focused on recovery. Even breathing affects brain states. Slow, deep breathing activates the parasympathetic nervous system, promoting calm. Rapid breathing increases arousal through sympathetic activation. This is why breathing exercises are effective for managing anxiety. You're not just calming your mind. You're actively modulating brain states through the brainstem circuits that link breathing and arousal. The integration extends temporally as well as spatially. Your brain now is shaped by your brain yesterday and will shape your brain tomorrow. Development is a continuous process, not stopping at maturity but continuing through life. Learning constantly rewires circuits. Aging gradually alters structure and function. You're not a static entity but a process, a pattern maintaining itself while constantly changing. This integrated, dynamic view of the brain has implications. It means you can't isolate one function and understand it independently. You must consider context, connections, interactions. It means interventions that seem targeted at one system, a drug affecting one neurotransmitter, for instance, will have widespread effects because systems are interconnected. It means the brain-body distinction is artificial. Your mental states affect your body. Stress impairs immune function. Depression increases inflammation. Positive emotions promote cardiovascular health. And your body affects your mental states. Exercise improves mood. Gut health influences anxiety. Chronic pain alters brain structure. Mind and body aren't separate. They're aspects of an integrated organism. This integration is what makes neuroscience both fascinating and frustrating. Fascinating because the interactions produce complexity and emergent properties that couldn't be predicted from isolated components. Frustrating because understanding one part doesn't automatically explain the whole. You can understand synaptic transmission perfectly and still be mystified by consciousness. You can map every connection and still not predict behavior. The brain is a complex system, and complex systems have properties that emerge from interactions rather than residing in individual components. Consciousness might be such a property, arising from patterns of interaction across billions of neurons rather than from any specific neural mechanism. If so, searching for the neural correlate of consciousness might be asking the wrong question. It might be like asking which water molecule is responsible for a wave. The wave is a pattern, not a thing, emerging from interactions. This doesn't mean consciousness is supernatural or non-physical. It means it's a property of organization, of how components relate rather than what components are. Understanding it might require not just neuroscience, but systems. Neuroscience. Studying networks and dynamics rather than individual neurons and molecules. We've covered enormous ground in this exploration of the nervous system. From gross anatomy to molecular mechanisms, from sensory processing to consciousness, from development to decline. But we've necessarily simplified. The real brain is vastly more complex, with details and subtleties that fill libraries. What should you take from this? That the brain is knowable. It's not magic, not mystical, not fundamentally different from other biological systems. It's cells, chemistry, electricity, following physical laws. But it's those laws applied at extraordinary complexity, producing functions that still astonish. Perception that constructs reality from patterns of neural activity. Motor control that translates intentions into coordinated action. Memory that encodes experience in synaptic strengths. Emotion that assigns value and drives motivation. Consciousness that generates the subjective experience of being. We understand the machinery increasingly well. What we're still puzzling over is how machinery produces experience, how objective processes become subjective phenomena, how the physical brain generates the mental mind. That gap remains, the hard problem persisting at the center of neuroscience. But perhaps the gap will narrow. Perhaps continued investigation will reveal that the mystery dissolves once we understand the details fully. Or perhaps consciousness will remain mysterious, pointing to something about nature we haven't yet grasped. Some principle beyond current physics and chemistry that governs how systems become aware of themselves. Either way, the journey is remarkable. From a sheet of cells folding into a tube, through precisely orchestrated development, to a structure capable of understanding itself, the brain is the universe becoming conscious of its own existence. You are the means by which the cosmos examines itself. And that, regardless of remaining mysteries, is worthy of wonder. The mystery remains. We've reached the end of our journey through the nervous system, though in truth, we've barely scratched the surface. The human brain, with its 86 billion neurons and 100 trillion synapses, contains more complexity than we could explore in 100 lectures, let alone one. But you've seen the landscape now. You understand the major territories, the key principles, the mechanisms that underlie everything your brain does. From the electrical signals racing down axons to the chemical messages crossing synapses. From sensory receptors transducing physical energy into neural code to motor neurons commanding muscles to contract. From synaptic plasticity encoding memories to the vast networks generating consciousness itself. The machinery is physical. That much is clear. Neurons are cells following the laws of chemistry and physics. There's no magic ingredient, no ghost in the machine, no special consciousness particle that makes neural tissue different from any other tissue. The brain is biology, electrochemistry, networks of cells communicating through action potentials and neurotransmitters. And yet, from this machinery somehow emerges you. Your thoughts, your feelings, your memories, your sense of being a self observing the world from behind your eyes. The sensation of red, the taste of coffee, the feeling of anxiety before a presentation, the warmth of recognition when you see a friend. These experiences, these qualia, are produced by neural activity. But how? Why? That question, the hard problem of consciousness, remains unanswered. We've mapped the correlates, identified the regions, described the mechanisms. We know that consciousness requires an intact cortex, that it emerges from integrated information processing, that it can be manipulated through drugs and electrical stimulation. But correlation isn't explanation. Understanding the machinery doesn't explain the experience. Perhaps future neuroscience will solve this. Perhaps we'll discover that consciousness, like life before molecular biology, is simply complex organization of ordinary physical processes, mysterious only until we understand the details fully. The gap between mechanism and experience might dissolve once we know enough. Or perhaps consciousness points to something we haven't yet grasped, some principle about nature that current physics and chemistry don't capture. Not mysticism or dualism, but a genuine property of certain kinds of complex systems that we don't yet have the conceptual tools to describe. Either way, the mystery shouldn't diminish the achievement. We've learned an extraordinary amount about how the brain works. We can restore vision to the blind with retinal implants. We can treat depression by modulating serotonin. We can help paralyzed patients control robotic limbs with brain-machine interfaces. We can enhance learning, slow cognitive decline, even grow new neurons through exercise. The practical applications of neuroscience have transformed medicine and will continue advancing. But beyond the practical benefits, there's something profound about understanding yourself, about knowing that every thought, every memory, every emotion is the product of this 3-pound organ sitting in darkness inside your skull, constructing reality from patterns of neural activity. You are your brain, not in the sense that you're nothing but neurons, but in the sense that your mental life, your consciousness, your sense of self, all emerge from neural processes. There's no separate you observing the brain from outside. The brain is observing itself. You are what it feels like to be a brain. This realization can be unsettling. It seems to reduce the richness of human experience to mere mechanism, but it needn't. A sunset doesn't become less beautiful when you understand the physics of light scattering. Music doesn't lose its power when you comprehend the auditory processing that converts sound waves into emotion. Understanding mechanism can deepen appreciation rather than diminishing it. Your brain right now is doing something extraordinary. It's processing these words, extracting meaning from patterns of light or sound, integrating that information with everything you already know. Perhaps forming new synaptic connections that will encode this knowledge into memory. It's monitoring your body, regulating your heartbeat and breathing, maintaining balance and posture. It's generating the continuous sense of being present, being you, experiencing this moment. And it's doing all of this through mechanisms we've explored. Neurons firing, synapses transmitting, networks coordinating across regions, plasticity enabling learning, integration producing unified experience. The machinery we've described isn't some abstract other. It's you, doing what you're doing right now. So, as you drift off, as consciousness fades into sleep, as your brain shifts into different modes of activity with different patterns and purposes, remember this. You are the universe becoming aware of itself. You are matter arranged in patterns complex enough to think about its own existence. And that, mysterious as it remains, is remarkable. The hard problem persists. Consciousness remains unexplained. But the brain, in all its physical, chemical, electrical complexity, is knowable. And knowing it means knowing yourself in the deepest possible way. Sleep well. Your brain has work to do tonight, consolidating today's experiences into tomorrow's memories, preparing you for whatever comes next. It knows what it's doing, even if we don't fully understand how. The machinery works, the mystery remains. And both are worthy of wonder.